भजेऽहम् सदाम्बोधिवासं गलत्पुष्पहासं जगत्सन्निवासं सदा दैत्यवासं गदाचक्रशस्त्रं लसत्पीतवस्त्रं हसच्चारुवक्त्रं भजेऽहम् रमाकण्डहारं श्रुतिभ्रान्तसारं जलान्धविहारं धराभारहारं चिदानन्दरूपं मनोज्ञस्वरूपं त्रिदानेकरूपं भजेऽहम् जराजन्महीनं परानन्दपीनं समाधानलीनं सदैवानवीनं जगज्जन्महेतुं सुरानीककेतुं त्रिलोकैकसेतुं भजेऽहम् त्रिलोकैकसेतुं भजेऽहम् त्रिलोकैकसेतुं भजेऽहम्
Now, I would like to invite Dr. P. Kalaiselvi Ma'am, Professor and Head Department of Pharmaceutical Analysis, JKK MMR of Anne JKK Samuel College of Pharmacy, to deliver the welcome address for the inaugural ceremony. Welcome, ma'am.
A very pleasant morning to everyone gathered here. It gives immense pleasure and great honor as the organizing secretary to extend a hearty welcome to each and everyone on behalf of our management in the Department of Pharmaceutical Chemistry and Pharmaceutical Analysis, JKK MMRFS, Annai JKK Samburniyammal College of Pharmacy, Kumarbalayam, to this one-day international seminar. Today's seminar brings together eminent academicians, researchers, professionals, and students have come together to share the knowledge, to exchange ideas, and explore the latest achievement in pharma science. We are truly privileged to have with us our esteemed chief guest, Dr. G. Muruganandam, Principal, Swami Vivekananda College of Pharmacy, Tiruchengodu, and our distinguished guest of honor, Dr. V. Sendil, Principal, Vivekananda College for Women, Sangagiri. We are deeply grateful your gracious presence and willingness to share our valuable knowledge and experience with us. We wholeheartedly welcome you for this seminar. Welcome, sir. Now I take this opportunity to extend my respectful and affectionate welcome to our chairman Amma, who's this visionary leadership and constant source of motivation. Welcome, Amma. To it my proud privilege to extend a very warm and respectful welcome to our beloved correspondent, Mr. JKM Jaiprakash sir, who always a pillar of strength and inspiring guide for all our academic and research initiatives. Welcome, sir. And also extend my warm welcome to our esteemed directors, Ms. Srinitya ma'am and Mr. Karan sir, whose valuable guidance and continuous encouragement to motivate us. Welcome, sir. With great pleasure, I extend my respectful welcome to beloved Principal Dr. N. Sendil Kumar sir. Your dynamic leadership and thoughtful guidance have enabled us to organize the seminar a grand success. Welcome, sir. A special and heartfelt welcome to all our distinguished speakers, resource persons who have come from various institutions and organizations to share their expertise knowledge. Welcome, sir. Welcome, ma'am. And also, I am very happy to extend a warm welcome to our organizing committee members and team members whose tireless efforts, dedication and teamwork have made this seminar meaningful. Next, I wholeheartedly welcome all our faculty members, delegates, research scholars and students who have joined from various zones in and around Tamil Nadu. Last but not least, I extend my sincere thanks to welcome our non-teaching staff members and student volunteers whose coordination and hard work have been instrumental in making this event successful. Once again, I extend my warm welcome to all the dignitaries, speakers, delegates, faculty members, students and guests assembled here. I wish this international level seminar a grand success and look forward a day filled with insightful learning, inspiring interaction and meaningful discussion. Once again, I warm welcome you all for this wonderful session. Thank you.
Thank you, ma'am. With great respect, I invite our principal, Dr. N. Sendilkumar sir, to address and greet the gathering.
Good morning everyone. So with the blessings of our Ayya, today we are organizing the international seminar from the Pharmaceutical Chemistry Department. So before that, respected our management, Karan sir, director of our institution, and my dear friends, Dr. Muruganandam, Principal of Swami Vivekananda College of Pharmacy, Tiruchengodu, and my dear friend, Dr. V. Sendil, Principal, Vivekananda College for Women, Sangagiri, and my dear friends, Ravichandran sir, Gomadi madam, and Parasuraman sir. They have come here for giving a wonderful seminar for this today's session. So first of all, I would like to welcome you all to our institution. Maybe some of you might have visited earlier, and maybe some of you are new to our institution. So we are welcoming you to our institutions. Of course, we have got a very nice response. There are so many colleges, more than 40 colleges. They have responded and registered as delegates. Really, it's a wonderful response from all the colleges and my dear friends working in other colleges also, and my dear faculties, and my dear students, and my dear delegates. So this seminar, definitely, as per the curriculum, we need to organize some seminars. All the colleges, not only our colleges, all the colleges have to organize this type of seminars, and we have to give some updates of our subjects and also syllabus. So these kind of seminars will be definitely useful for your future career. We have to listen and we have to get ideas, and there are eminent speakers here today. They will give a lot of ideas regarding the subject and also for your future career. Use it. Definitely it will be very useful and utilize it. And also, this is a chance to interact with all other college friends, isn't it? So in this seminar, not only we are attending the seminar, and also this is a chance to interact with the other college students. So this is a very good platform, and likewise you should always try to attend the seminars wherever whatever the colleges are conducting the seminars. Please try to attend the seminars and also interact, and so that you will get a lot of ideas for your future scopes. So of course our pharmacy profession having a lot of scopes for your future. Still a lot of demands are there. this is our profession is the only profession still not yet saturated. So still demands are there and opportunities are there. இல்லையா? யாரும் நம்ம ஃபார்மசி படிச்சுட்டு வேலை இல்லைன்னு இல்லை. அதேமாதிரி we can get a lot of very good jobs. அதேமாதிரி promotions, growth, achievements. We can do a lot of achievements in our profession. So I think luckily you have chosen this profession for your future career. Definitely it will be helpful for your future. So use it, utilize it, the chance, and interact with the speakers. If you have any doubts, please interact with the speakers. Okay? And my humble request, during the session, don't make noise here. சரியா? செஷன் நடக்கும்போது கண்டிப்பாக கவனிக்கணும். கவனித்தா தான் புரியும். சும்மா வந்து உட்காந்துட்டு கதை பேசிட்டு போகக்கூடாது. பேசிகிட்டு இருக்காதீங்க. அப்புறம் வெளியே போய் பேசிக்கலாம். சரியா? அதனால் வந்து ட்ரை டு லிசன். See, there are experts from our field. They have come here to give ideas, guidance for your future life. Utilize it. அதனால் அமைதியாக வந்து கேட்டுக்கோங்க. அப்புறம் போய் பேசுங்க. என்ன லஞ்ச் டைம் இருக்குது, பிரேக் டைம் இருக்குது. அந்த டைமில் நீங்கள் you can interact with other college students also. And of course, you have arranged classrooms also with the video and audio. We have arranged classroom also because of the crowd. To manage the crowd, we have arranged parallelly the classrooms also. You can sit inside and you can watch. Sorry, Anala, don't waste the chance. Okay. So with these few words, thank you very much, and enjoy the day. I hope this will be very useful for your future career. Thank you very much. Thank you.
Thank you, sir. Now it's time to felicitate our respected dignitaries. I kindly request our principal sir to felicitate our esteemed director of institution, Mr. Karan Raja sir. Thank you, sir. Now, I request our respected principal sir to felicitate our chief guest, Dr. Muruganandam sir. I request Mr. Karan Raja sir to felicitate our chief guest, Dr. Muruganandam sir. Thank you, sir. I request our Mr. Karan sir to felicitate our guest of honor, Dr. V. Sendil sir. Please. I request Mr. Karan sir to felicitate our eminent speaker, Dr. V. Ravichandran sir. ma'am to felicitate our eminent speaker, Dr. Gomati ma'am. I request Mr. Karan sir to felicitate our eminent speaker, Dr. Parasuraman sir. Thank you, dignitaries. It is my great pleasure to welcome our esteemed chief guest, Dr. G. Muruganandam sir, Professor and Principal of Swami Vivekananda College of Pharmacy, Trichangodu, with 27 years of teaching and research experience in the field of pharmacy. He has made significant contribution to pharmaceutical education through 2 Indian patents, 41 research publications, a book on biochemistry, and several book chapters. He also organized many national and international conferences and currently guiding PhD scholars. He also a Senate member of the Tamil Nadu Dr. MGR Medical University, Chennai, and a life member of Association of Pharmaceutical Teachers of Tamil Nadu branch. We are truly honored to have our chief guest, Dr. G. Muruganandam sir, to deliver few words. Welcome, sir.
A very good morning to all gathered here. Respected Karan Raja sir, Director, JKK Muniraja Institutions. My teacher, beloved principal, many words more to add. Dr. Sendil Kumar sir, guest of the honor, Sendil sir, distinguished speakers on the stage, eminent teachers off the stage, students and friends. Today is one of the important day in your life because some of you may get triggered to carry out some research. The main importance of organizing conference is just not giving you some idea or exchanging some thoughts. Here are the people who is going to share their hands-on experience in the world of AI, artificial intelligence. By the click of the button you get everything, but still there is something called experiential learning which will be shared by the eminent speakers. So all of you make use of this particular platform, interact with them and learn something new in your life, because you know very well education is everywhere, everyone is offering degree, but what you need to add in your career is something called value. A student, when they are learning along with the education knowledge, who adds additional value will be definitely a successful person in his or her career. So with these few words, I really appreciate all the organizing committee members, faculties of JKK Muniraja for bringing such a wonderful speakers to the conference. Actually, a conference success is mainly based on the speakers, because today scenario, whatever the venue you organize, wherever you organize, if the students are not happy with the speakers, you don't get registration at all. So in that aspect, our Sendil sir and organizing secretary and the entire team, they have done a wonderful job. So I wish them all the best. I also request the management to support all the teamwork here. And I know very well there will be a lot of student volunteers might are involved in this organizing this conference. So I wish each and every one of them all the best. And all the delegates, I welcome you to the conference. And as already our Sendil sir informed, please listen carefully. Next 3 hours of the interaction with the speakers is very important. I wish you all the best. Learn something, adopt in your life, and add value. Thank you so much.
Thank you, sir. It is my privilege to introduce our guest of honor, Dr. V. Sendil sir, Principal, Vivekananda College for Women, Sangagiri. He is a passionate and excellent researcher and an inspiring mentor who has dedicated his career to advancing pharmaceutical sciences. He has around 30 years of experience in teaching and research, with over 80 research publications, 2 Indian patents, 2 authored books, and the successful guidance of several PhD scholars. He has made a significant impact on pharmaceutical education and research. Today, we are truly fortunate to have such an eminent scholar and inspiring personality with us. I request our esteemed guest of honor, Dr. V. Sendil sir, to kindly deliver a few thoughts. Welcome,
you.
sir.
A pleasant good morning to one and all. So today is the one-day seminar organized by the Department of Pharmaceutical Chemistry, JKK Muniraja College of Pharmacy. So first of all, I would like to thank the management, correspondent, director, principal of this esteemed institution, organizing committee, and faculty of pharmaceutical chemistry in JKKM. So today the evolution is started. The last 4-5 years, the AI era. Before that, the advanced drug delivery system, and 10 years back. The last 10 years, we are using the advanced drug delivery system. But last 5 years, we are using the AI integrated. So artificial intelligence is capturing the most of the area, like drug delivery system, healthcare system, automobiles, agriculture, like various fields. The artificial intelligence are making a big impact. So, as a student, you want to learn how to utilize artificial intelligence in pharma field. That is the way we have to develop yourself. Because who develop the skill in artificial intelligence? So, what are the applications are there? You want to utilize properly. So artificial intelligence will not a destruction of your life. It will be develop your life. So what are the applications are there? How to utilize the artificial intelligence? How to effectively use to develop the drug delivery system? So rightly selected the topic, green synthesis. So what is a green synthesis? So when we are student, like 20-30 years back, for any projects, we are used a lot of organic chemicals, and we are using a lot of energies for developing any drug or synthesis of any drug. But now we are insisting the green synthesis. So green synthesis is what is the natural source, like water. We have to use the water for extraction process, for reaction process. So how to effectively use the water and how to use the energy like ultrasonics, microwaves for synthesis. So these are the natural sources, how we have to effectively use for the synthesis. We have to reduce the sources, what it is costly, that will be make the impact or pollution to the environment. That we want to look into that. So without any environmental pollution, without any noise, we want to use the synthesis and effectively with the low cost, with less human power. So that is the green synthesis. Okay. Next is the docking. So what is the docking? You know, now a lot of softwares are there, like Autodock, Autodock Veni, MOE and Salinger. Like so many softwares are there. So what is the use of that? So that softwares are like key and lock. So lock here is a protein and key is the molecules, a ligand or ligand with any drug molecules. So which drug molecules will actively bind with the proteins? So that today docking is most of the eminent speakers are going to teach like how to find out the suitable protein and how to lead components to attach with that. So what is the binding capacity and what is the how long it will be react. So these things and all going to explain. So here three eminents are present here. So you also to develop in particular field in future. In the particular field you have to develop in a particular area. So then only you will come and sit in this stage in future. So here lot of opportunities are there. You have to select which area is very important, which area next 10 years going to dominate. So that area you have to select and you have to develop yourself. Then only the stage is yours. Otherwise, you will not occupy the stage. If you want to occupy the stage, you want to develop yourself. So this is the take-home message. And with low energy, how output? Because the next 10 years, the sales, the growth of pharma field is 2 trillion. 2 trillion means 2 lakh crore. So just imagine it. So after the China, India is a major producer of API. So what is our input for developing our nation? So that you have to think and you have to act accordingly. So what is the next 2 to 3 years? Next 2 to 3 years, what is the AA? So AA is the learning management system. Is one of the part. The Google or Gemini AI going to develop shift to their platform from Alpha 3 to Alpha 4. So now we are using is the Alpha 3. Next is the Alpha 4. Very deeply they have to sensorize the particular area and they have to categorize different type of proteins and the molecules which is suitable. That is also deep learning the AI will be give you. So the technology is developing where you are, how you are going to effectively utilize that. That's very important. So dear students, just you have to think it and what is your future and what is the next 4 to 5 years, what is the changes will happen, you just forecast it. You just change yourself and you have to move fast and work fast and you have to achieve something. So forecasting is very, very important. So the existing development and next 4 years, what is the developing stage? So you just think it and you have to develop yourself and shape it yourself. Then only you will achieve something. Thank you for giving the opportunity and thanks for organizers and please use this platform very effectively, all the students who gathered here. Thank you.
Thank you, sir. Learning gives creativity. Creativity leads to thinking. Thinking provides knowledge, and knowledge makes you great. It's now time for the official release of International Seminar Abstract Book. We request our dignitaries to kindly release the abstract book. request Dr. E. Tilagam Ma'am, Vice Principal, JKK MMR of Anne JKK Samunimal College of Pharmacy, to deliver the vote of thanks.
Good morning to all. Esteemed dignitaries, distinguished speakers, respected organizer, dear participants and valued guests. It is indeed a great honor and privilege for me to deliver the vote of thanks on the special occasion of our one-day international seminar on greener pathways to better medicine, eco-friendly synthesis, molecular docking in modern drug development and analytical technology at JKK MMRF and JKK Samunimal College of Pharmacy. I take this opportunity to express my heartfelt gratitude to our chief guest, Dr. Muruganandam sir, Principal, Swami Vivekananda College of Pharmacy, Tiruchengood, for graciously accepting our invitation and being with us today. Your inspiring address has enlightened us and your presence had added immense value of this seminar. Thank you, sir. I express our deepest gratitude to our chairman, Srimati Vasanta Kumari Muniraja Amma, and correspondent, Mr. J.K.M. Prakash sir, J.K. Muniraja Educational Institution, for their constant encouragement, guidance, and support which made this seminar possible. Your vision and leadership continue to inspire us in all our academic endeavors. Thank you, Amma and sir. My sincere thanks also extend to respected directors, Ms. J. Nityasri Madam, Mr. J. Karan Raja sir, for their encouragement and have greatly contributed to the successful organization of this international seminar. We deeply appreciate your commitment to academic excellence and overall growth and progress of our institution. Thank you, sir. A special word of appreciation goes to our respected principal, Professor N. Sendil Kumar sir, for your continuous support, valuable suggestions, and your active involvement has been a great motivation to all of us. Thank you, sir. We are also immensely thankful to our guest of honor, Dr. V. Sendil sir, Principal, Vivekananda College for Women, Sangagiri, for joining us today and sharing their valuable thoughts and experience. Your presence has truly enriched our event. Thank you, sir. I extend my deep appreciation to all the invited speakers and resource persons who have taken time from their busy schedule to share their knowledge and expertise with us. Thank you, sir, and thank you, madam. A special thanks to the faculty members and organizing committee members for their meticulous planning, tireless effort, and teamwork for this seminar. Without your dedication, this event would not have been possible. I also extend my gratitude to all the non-teaching staffs and technical support team for their coordination behind the seminar. To our student volunteers, thank you for your enthusiasm and dedication, which helped ensure the smooth flow of today's seminar. A special word of appreciation goes to participants and teaching faculties of various institutions for your enthusiastic participation and for upgrading this seminar with your knowledge and perspectives. With a heartfelt gratitude, I once again thank you everyone who made this seminar a meaningful and memorable one. Thank you one and all. Thank you.
Thank you, ma'am. Dear delegates, kindly note that the oral presentation will be conducted on first and second floor and the e-poster presentation will be conducted on second floor. Our volunteers will guide you. The first session about to start. I request Dr. G. Muttububadi sir, Professor, Department of Pharmaceutical Chemistry, Vivekananda College of Pharmacy for Women, Sangagiri, to kindly address today's eminent speaker, Dr. V. Ravichandran sir. Welcome, sir. I request everyone, please be seated. Delegates, please be seated. I request, dear delegates, please assemble to the seminar hall.
Very good morning to all. It is my privilege to introduce our esteemed resource person, Professor Dr. V. Ravichandran, M.Pharm PhD, FSAB. Professor Dr. V. Ravichandran is currently serving as Professor, Head of the Pharmaceutical Chemistry Department and Deputy Director, Center for Postgraduate Studies at AIMST University, Malaysia. He has over 27 years of distinguished experience in teaching and pharmaceutical research. He is by Stanford University and elsewhere, and is also ranked among the top 1,000 scientists in Malaysia. He has received several prestigious national and international awards for excellence in the teaching and research. With great pleasure, I invite Professor Dr. V. Ravichandran to deliver today's resource session. Sir, we are honored to
Hello? Can I start? Okay. Think it's okay. No, it's okay. Okay. Recording adalla. Good morning to all, respected dignitaries, fellow colleagues, professors, lecturers, and my dear participants. So it's a great pleasure to be here today to share my knowledge, whatever I know about the eco-friendly synthesis. So many of you already heard about these things, about eco-friendly synthesis and docking studies. Even though I want to share what I know. Yes, I'm working in this field the last 25 years. In the year of 2000 onwards, I'm working in this field in different direction. Like the next topic you are going to hear, the molecular docking. That is also one of the way for the eco-friendly synthesis. It is not directly involving, but it indirectly helpful to do the eco-friendly synthesis. So let me start with, begin with a simple question. I want to ask one question. Okay? So we are pharmacists, so I'm going to ask question in a pharmacy field. So can we develop an effective pharmaceutical compound without compromising the health of planet? Can we develop an effective pharmaceutical compound without compromising the health of planet? Can or cannot? Those who are telling can, just raise your hand. என்ன கேட்குறேன்னு புரியுதா? So we want to develop a pharmaceutical active compounds. So without affecting our planet. Is it possible or not? Can or cannot? Can. Anyone is telling cannot? Or both? Just raise your hand or tell something. Whether it is can or cannot or can and cannot. Okay? It is both. Very good. It is both. Either can or cannot. Means what the way we are going to do the things. That is one of the things, if you are going to follow eco-friendly synthesis, then it's can. If you are following the old synthesis, that is the conventional method, then it's not possible at all. It is definitely going to affect the environment. So what is the conventional method of synthesis? Do you have any idea what is conventional method of synthesis? So where we are going to get more yield, that is my main target. Why we want more yield? To fulfill the need. Why the need is rising day by day, population. So we are not controlling the population, so the need is rising, so automatically we have to produce the required quantity. So we are following the conventional method, which is giving lot of yield. So what is the problem of conventional method? If you know that, then it is easy to move to the eco-friendly synthesis. What is the problem of our conventional? What you are doing in your laboratory, in the chemistry lab? You are synthesizing lot of compounds. How you are doing? Just taking A and B, mix, heat, reflux, and you are getting the product. And you are filtering and recrystallization. Whatever the things you are doing, you are finding out the percentage yield. So this is the things in the industry also they are doing in the bulk quantity. What is the problem in that? The waste. The main thing is the chemical waste. There is lot of waste produced by conventional method. The second thing is we are using lot of hazardous material. The third thing, we are using the toxic solvent. So when you are using that petrochemical related solvent like benzene, chloroform, diethyl ether, so when we are heating, it is evaporating. Evaporating means it is mixed with the atmosphere. It is spoiling the atmosphere. So these are the few major things. Chemical waste, that is the first one. The second one is hazardous substance. Third one is toxic solvent. And the other important thing is we are using intensive energies. By heating, we need the energy. Or by cooling, we need the energy. Or electronical heating, we need the energy. So everywhere we are using the intensive energy, which is non-renewable. So only these are the drawback of conventional method. So those things we have to minimize, then it is called as eco-friendly synthesis. That only we are going to see today, how we are going to minimize all these things, the chemical waste and other things. Do you have any idea what is the wastage produced by pharma companies in a year, in a industrial wastage? How many percentage? The total industrial wastage is there in the world. In that, how much it is produced by pharma company? Very good, it is nearby, 18%. 18% it is produced by pharma company itself. So, if we are using the chemical waste, if you are using the toxic solvent, if you are using the hazardous material, what will be the problem if you are using this? So the chemical waste, what it is going to do? It is going to harm the environment. It is going to increase the environmental pollution, in environmental burdens. When the environmental pollution is there, then what will be the further effect? It will affect the sustainability. That is the main thing what I am going to discuss today, sustainability. If the sustainability is affected, then it is going to affect. Next. Yes, human health and animal health. When the sustainability is checked, then automatically it is going to affect the human health, animal health, whatever it is. It is a circle process. So we have to plan the process where we have to reduce all these things. Instead of once I done the synthesis, then after getting the wastage, don't think that how to clean the waste, how to destroy that waste. Instead of that, in the beginning itself, when you are designing itself, you have to design without wastage. That only called as eco-friendly synthesis. So now, I think really I am surprised when I entered your principal room. Have you ever noticed from here the lecturers and the students of JKKM, I think. Have you noticed anything different in your principal room? Really, I have gone throughout Tamil Nadu, more than 20 colleges I visited. Throughout India, it is more than 50 colleges. I never seen that in any principal room. Today, I have seen that one. Do you have any idea what is that? Any idea? The one college students or the lecturers. Really, I am surprised. If you noticed that one, then this is nothing. There are 17 goals are there. Sustainable development goals. It is just near to his right hand or left hand. He has kept the big banner like that things. The 17 goals are there. So we have to achieve that goal. That is developed by United Nations. Near about 53 countries, they accepted these things in 2012 and implemented in 2015, and we have to reach all the 17 goals in 2030. Even our Prime Minister also giving full support for that. All the government, not only India, Malaysia, many of the 53 countries, they are together to achieve that 17 goals. Later we will see what are those 17 goals. Okay, that is the really so now the sustainability, just now we talked about sustainability. What is sustainability? So sustainability now is no more option. Now we have to follow, it is compulsory. Because now the season is monsoon season, I think. Why we are not getting rain? Why there is no water in the river? What is the reason? Who is the reason? We are blaming each other, politician, ministers, or the government officials. Who is the reason? We are the reason. First, you have to accept that each and everyone is the reason for what we are facing today. So think about our next generation. Because I didn't think, so you are suffering. Now you are not think, then definitely your next generation is going to suffer more than this. Even they will not get drinking water or food. So that is the main reason we have to concentrate this one. I think I really appreciate the management to select this topic. Okay? So that is the reason. So now sustainability is no more option. We compulsorily, particularly in pharma industry, we have to follow that one. It is compulsory. So the main motto of my today talk is this one. Little or no harm to the earth. Particularly special emphasis on our pharma field. That is the main thing. Then here you see I have given the subtitle Advancing Sustainable Pharmaceutical Research for Your Green Future. So we are going to use the advanced techniques which is related to sustainability in the field of pharmaceutical research to make our future green. So these are the things what I am going to discuss today. Okay. So as a human, we are not, anyone is not perfect 100%. Everyone will do a mistake. Same thing. Human activity is completely risk-free. Whatever we are doing, there is a risk. Suppose I want to go from here to E-Road. I am going in bike. There is a lot of risk I have to face. If I am operating about the risk, then I have to sit here only. I cannot go. I cannot learn or earn whatever it is. So everything is risk is there. So use common sense. That is the first thing. How we are going to use common sense? Suppose I want to go to E-Road. I am alone. I am having enough time. Then I will prefer bus. If some emergency, then I have to go by bike. If it is raining, then I am having the car, then I can use the car or I can call the taxi and go. So you have to use your common sense. So no need to simply waste the time and money for unnecessary things. The next one is chemical sense. So whenever you are doing some synthesis, some unwanted material will be there. There is no need to use that. So you have to apply your common sense, that one. Then follow the rules of life safety. That is the most important thing. Because in sustainability, there are three things will be there. Cleaner, safety and energy. Use renewable energy. These three things are important when you are talking about sustainability. So, chemistry. So, tell any one things in the world without chemistry. Anything, it is direct or indirect way, it is related with chemistry. So, we cannot tell chemistry is 100% useful. The same way it is having the side effect or other effect. That is, so the other effect, side effect or adverse effect, something. So whenever you are using the chemistry, you have to take care about that also. Suppose if you are any industry, you can take not only the pharma industry, you can take any agricultural industry, or you can go for engineering industry, biotech industry, everywhere the problem is there. Because of the chemical atoms. So it is going to pollute the volatile organic compounds, that is mainly we are using in pharma industry. Greenhouse gases. What is greenhouse gases? What is the role? Why it is called as greenhouse gases? What are those? Example for that? Very good. Carbon dioxide, methane, nitrous oxide, ozone, nitrofluorine gases. All it is called as greenhouse. Why it is called as greenhouse gases? Any reason? Because it is used to control our climate. We know that carbon dioxide, it is always giving the problems. No, it is controlling our climate. When the level is increased, then only the problem will come. So automatically in the environment, it is in the optimum level. We are the one, we are increasing the level of carbon dioxide, methane, all the greenhouse gases by using the petrochemicals like coal tar, gas, oils like petrol, diesel, all it is producing all these things, methane and carbon dioxide. So we are the one. So that also we have to take care. Then ozone layer depletion and aqueous pollution. Directly or indirectly we are responsible. Okay. So industrial and urban waste, fertilizers and so on. Then solid pollution. So here I didn't mention about our pharma industry. So just now I told the pharma industry is the major industry which producing near about 18% of the industrial waste throughout the world. So the global challenge. So pharmaceutical manufacturing generate 100 kg of waste to produce 1 kg of API. API, I think you all know, active pharmaceutical ingredients. They want to produce 1 kg of the compound, you have to produce 100 kg. So the E factor is very high, 25 to 100. Now what is E factor, environmental factor? How to calculate? Because in green chemistry, we should know some metrics also. One of the metric is E factor. So we have to calculate the E factor. The E factor should be the minimum. But in pharma industry, it is 25 to 100. Means we are producing, the formula is the total wastage in kilogram divided by the mass of active pharmaceutical, the final product in kg. So 100 divided by 1. So here 100 gram waste divided by 1 gram API. So the E factor is 100. So how to solve this? So these are the solution. First you have to do market imperative towards the green chemistry. Then there are a lot of regulatory is there. One is ICH guidelines, then Environmental Protection Agency, then European Medical Agency. These are the some agency who is controlling this. And the strategic shift. Just now what I told, we have to move from conventional method to eco-friendly method. Then this is the focus. Means we have to focus on the principle, the methodology, the metrics. Just now I told one of the metrics. So these are the few things we have to concentrate more to bring eco-friendly synthesis. This is the one what I told I have seen in your principal room. The 17 goals, which was introduced by the UN, United Nations. 53 nations, they agreed already. So in this, where our pharma field is going to influence in this out of 17. Just go to the number 3, number 6, number 8, number 9, number 12, 13, 14, 15 and 17. These are the 9 areas where we can influence as a pharmacist. 3, 6, 8, 9, 12, 13, 14, 15 and 17. Either directly or indirectly we are going to play a role. So these are the 9 areas, the 9 principles where the pharmacists do the changes. The next one, the green chemistry, it is having 12 goals. So this is the basic principle of the sustainability. From prevent waste, atom economy, less hazardous chemicals, design safer chemicals, safer solvent, design for energy efficiency, use renewable feedstock, reduce derivatives, catalysis, design of degradation, and real-time analysis for pollution prevention and to prevent accident. So these are the 12 areas where we are going to see, means now what you are going to see, this is the 12 principles. So the green chemistry is about to reduction of waste intensification and catalytic paradigms. These are the three areas what we are going to see. In the metrics, we are going to see E factor, just now we have seen. Then PMI, that is process mass intensity, solvent assessment. Then process intensification, continuous flow chemistry, micro reactors. And the catalytic, instead of normal catalyst, just now I told we can use sulfuric acid. Instead of that, think which one is it is not harmful. Go for biocatalyst. You can use some enzymes. You can use some vitamins, amino acid to do the proceed the synthesis. Or you can use organocatalysis to proceed the reaction. So the environmental footprint of pharma, while fine chemical means the chemical which is used in the pharma industry. It is pharma chemicals or fine chemicals, low tonnage relative to petrochemicals. But when you are talking about the wastage wise, this will be the more wastage produced by fine chemicals than the petrochemicals. So the pharmaceutical industry emits more carbon dioxide equivalent per $1 million of revenue than the automotive sector. So automotive sector, you know, the automobile industry where we are using lots of petrochemicals. Even that is producing less wastage, less carbon dioxide than pharma industry. The pharma industry is producing more carbon dioxide. So now you can understand what is the importance of implementing the green chemistry, the 12 goals. So heavy solvent usage in pharma industry, high organic waste, greenhouse as intensive, then low structural atom utilization. Why we are using a lot of the hazardous chemical or the toxic solvent? Because even we are going to use a small molecule, but when you are seeing the complexity, it is more complex than any other chemical. Whether it is a petrochemical or all other chemicals, our pharma products are having complex structure. That is the reason to make that complex structure, we need lot of steps to proceed to get the particular compound. That is the reason why there is a lot of wastage. So the other important problem in the conventional method, we are doing in batch wise. Because the bulk we are doing, that is one of the drawback. When you are doing in the bulk, you are going to take a big vessel. In that, kg of ingredients will be added and you are going to heat or stir, whatever it is in the bulk quantity. Suppose if you are heating the big container, the heat will not be, the thermal will not be distributed equally. Instead of that, you have to go for the continuous flow process. The recent process is continuous flow synthesis. So instead of big vessel, you have to do the synthesis continuously by using your small tubes. So in the tubes, our ingredients will move. So here heating, next cooling, then filtering, everything will be done in the flow. So it will be very fast and the distribution, the thermal distribution also will be fast. Then just now we have seen the 12 principles. So apart from that, we want to implement the sustainability or eco-friendly synthesis. So these are the eco-friendly synthesis demands vigorous industrial parameters. These are the some industrial parameters what you have to see. So you have to analyze from the beginning and the economic parity, because I want to implement the eco-friendly synthesis. So it should not affect the cost of my product. If the cost of the product is increased, API, then nobody is going to buy. So automatically it is waste. So that also you have to keep it in mind. That is also important thing. Then the safety profile. You cannot compromise with the safety profile. That is the sustainability main rule is we have to produce clean, safer compounds, but we should not compromise with the quality and the safety of the product or efficacy of the product. That is the main goal of the sustainability. So next one is process mass intensity. Because already you have done in your lab, you have synthesized some compound in organic chemistry or medicinal chemistry lab. So finally what you did, you find out the percentage yield. So it is really assessing parameter? No. Just we are taking which one is the major starting material and the final product. including water, solvent and catalyst. Everything should be included here and divided by the mass of final API. So what is the difference between the yield what you are getting, the practical yield in your lab and this one? It is completely purified, means which is eligible to take. It is a medicine. So that much you have to do the purification. Then only the correct efficiency will get. So that you have to calculate. So the efficiency should be, when you are calculating this PMI should be less than 20. Means if the total product what I am taking is 100 and my final product should be 20, then only I will get 100 divided by 5, then only I will get 20. So when I am taking all the ingredients, all together it is 100 gram. My product is 5 gram. Okay, then it is PMI is okay, acceptable. But it should be less than that. So next E factor, just now I told it is calculated by the total waste in gram divided by the mass of final product. So it is the another important matrix to find out the efficiency of the synthesis. So here I have given the oil refining industry, the E factor is 0.1. And bulk chemicals 1 to 5, but you can see the pharmaceuticals. It is 25 to 100. Just now I told how we are calculating this one. So the E factor is very high. Means the wastage is more. Then atom economy. So how we are calculating atom economy? It is a theoretical matrix. How we are calculating the atom economy? What are the main ingredients? We calculate the percentage of atoms present in that, and what is the percentage of atom in the product? No, it is not correct way. So only here I have mentioned it is not the correct way. So you have to consider some other scales also, like echo scale and life cycle scale, because the reaction, there are a lot of other things are there. We are purification, we are doing recrystallization. There also we are using the solvents. It is not considered by this atom economy method. So that is also important thing. Then the massive footprint, means the impact is from solvent. So whatever we are doing in the pharma industry, we are using 90% of the things are non-aqueous things. Non-aqueous mass means the organic things. So how we are going to reduce that? So for that, these are the some solvent what we have to use. Preferred solvents are water, ethanol, isopropanol and so on. But we should not use these things. Benzene, carbon tetrachloride and so on. And restricted solvents, dichloromethane, dimethyl formamide and all these things. Because of according to the toxicity. So the alternative solvent, suppose I don't want to use or I am not getting the expected yield by when I am using water or ethanol or methanol, I am not getting the yield. Then what is the alternative? So you have to move to the alternative solvent, that is ionic liquids or deep eutectic solvent. You can use any one of this. So what is the advantage of ionic liquids? It will give thermal stability, it will give recyclability, tunable solubility and low volatility. And this one is used in different field. So we can see the difference here. So the ionic liquids are salt, which having the melting point of below 100 degree, that is ionic liquids. The deep eutectic liquids mixes are, it is a Lewis or Bronsted acid and bases, or it's a mixture of that. So the advantage of this one, deep eutectic thing, is we can easily change the polarity. So according to the requirement, we can change the polarity. So that is the advantage of the deep eutectic mixture. Next, supercritical fluid. What is supercritical fluid? If we can take some compounds, when it will reach its optimum critical temperature and pressure, it will behave as gas as well as liquid. It is acting as the diffusivity is like gas, but the density is like liquid. So it is having the both the property. The best example is carbon dioxide. Supercritical carbon dioxide. So when We can use this one for lot of purpose. We can use for extraction, we can use for purification and the reaction also. So when you are using this one, the toxic solvent residue in the processing is 0%. Meanwhile, the recyclability is 99%. That is the added advantage of when you are using the supercritical fluid. Then water phase synthesis. So we all know that water is cheapest one and easily available one. So we can go with this one. But when we are using water alone, it's not sufficient to carry out the sample which is soluble in organic solvent. Suppose I'm doing the reaction, both the reactant are not soluble in water. Then how to do the reaction? So I have to go to the organic solvents. But I don't want to use organic solvents because it is toxic in nature. So what I have to do? I have to use the surfactant to make the substance soluble. So that only here. Use the modern surfactant strategies to reduce the surface. So it will be easily entrapped with water molecule and it will be soluble. So reaction run over water suspension because of the interfacial hydrogen bonds, or you have to use any amphiphilic surfactant self-assemble in water forming hydrophobic pockets or low temperature. So the micellar technique, I think already you have studied in your physical pharmacy and other things, the micellar techniques, it is having the both head, hydrophilic head and lipophilic head. It will form as your micellar form. So one joint, it is joined all the hydrophilic, all the lipophilic head will be outside or inside. So that one, so the example, best example is this one, tocopherol polyethylene glycol succinate. That is one of the polymer, that is 750M is your design surfactant engineered by Lipsatz and co-workers specifically for organic synthesis in water. Suppose you want to do the organic synthesis in water, when the water is not the solvent, then we can use this one, TPGS 750M. So it will make the reaction. So it is constructed from tocopherol, that is nothing but vitamin E, polyethylene glycol and succinic acid. So these micellar nanoreactors concentrate organic reactant to 2 molar. So already I told the micellar will be like that. So it will bring all the compounds inside within 2 molar concentration. So the reactant will be very fast. So the example for this one is Suzuki-Miura cross-coupling reaction in water by using this one, TPGS. So the reaction is coupling of sterically hindered aryl halides with boronic acid can be achieved at room temperature. So both the things are soluble in organic solvent, but we are not using organic solvent. Here we are using the water with the help of the TPGS. It is, we make it soluble and the reaction will take place, particularly the boronic acid. So when we are doing this reaction, the high local concentration of palladium, you can see the catalyst loading. When you are using the traditional method in DMF, the palladium loading is 1 to 5 mole. But here you can see it is 99% less than that, 0.1 to 0.5 mole percentage. So next, how to do the reaction without using the solvent. Now we have seen, what we have seen? We have seen the alternative solvent. Ionic liquid or tape eutectic liquid, everything is alternative solvent, other solvent. But now what we are going to see is without solvent, how to do the reaction. Is it possible? Yes, it's possible. By using physics, means the mechanochemical properties. What is mechanochemical properties? Here I have not given. It is by using the friction or the shearing force. By using that we can make the product. That is this one, solid state and activation technology. So for that we are going to use all this technique like Paul mill, planetary mill, vibration mill, attitutor and E-pins mill and rolling mill. Just we have to take substance and grind it by using mill, or you can, for example, in our laboratory for first year students we are doing one of the synthesis, the skip base. I think already you have heard skip base. one aldehyde will react with amine, you will get the skip base. So for that, you have to take the organic solvent, you have to reflux for 1 hour, 2 hour, but instead of that, 15 minutes we can finish the synthesis. Just take one aldehyde and amine, just you have to add 1 or 2 drops of the lemon juice. Just grind it for 15 to 30 minutes, you will get the product. The percentage purity is more than 95%. But when you are doing the organic synthesis, you will get the yield also less, 30 to 40%. The purity is 50 to 60% only. But when you are using this method, you will get 90% yield and 90% purity. So that is the added advantage when you are using this method. But it's not applicable for all. So only in the beginning itself, you should have the chemical knowledge. That is most important thing. So mechanochemistry foundation. So we are going to use these 3 processes: friction, impact or shearing force. So this is the one what is happening when you are grinding in your mortar, the friction between your mortar and pestle. When you are grinding, the friction will generate that heat, the reaction will take place when you are grinding. So this method utilizes high energy ball mill, that is planetary mill. Metal or ceramic mill collide inside a vessel according to solid reagents. So by eliminating liquid phase solvents entirely, we avoid solubility limitation and solve massive waste challenges. So when we are using solvent, then only it is going to produce lot of waste. So whenever we are not using the solvent, we can reduce the waste, chemical waste. Then solvent-free API synthesis. Example, in crystal formulation, generating stable, highly bioavailable drug co-crystals in a single solvent-free steps with complete atom conversion. The next one is some amidation reaction. So amide, how you will prepare? You will react amino acid with amine. Then only you will get amide. That we can do it with some toxic, normal method we are using some toxic coupling agent. But here there is no need. Then metal catalyst, no need to use the metal catalyst. So one of the example is the sildenafil intermediate, one of the main precursor for the synthesis of sildenafil. Already you know what is sildenafil. So the synthesis is the pyrrole core. You have to synthesize the main core of the molecule is the pyrrole. So for that we can use this method just now what I mentioned. So milled with catalytic amount of silica gel as a solid phase dispersant. So we will get the product. So you see the isolated yield in solid state coupling is 98% and solvent record is 0%. Because in this process completely bypasses chlorinated solvent. Because when you are using the conventional method, you are using the dichloromethane to purify the compounds. So here there is no need of that. That is the added advantage. The next process is twin screw extrusion scale. So when you are doing the ball mill, it is again the batch mode. So already I told what is the drawback in batch mode. When you are doing batch mode, the thermal distribution will be very low. So when the thermal distribution is very low, the reaction will be getting down. So when the reaction is not completed, then the product is not pure product. So lot of waste will be produced. Then microwave assisted synthesis, that is the another important things, how we can implement the eco-friendly synthesis by using microwave. So what is the advantage of microwave assisted synthesis, organic synthesis? When you are heating normal, when you are taking in around bottom flask heating, the heat is not distributed equally because it has to cross the vessel, whatever you are taking the vessel, it has to cross the membrane, then the temperature will go inside. So the solvent which is surrounding the vessel will heated faster than the middle of the vessel. So that is the drawback. But when you are using microwave, the heating is throughout. It will be complete heating will be there. So you will get excellent product compared to the conventional method. In microwave synthesis, they are using the electromagnetic radiation with 2.45 kilohertz. Then it is mainly used in heterocyclic synthesis because whatever the products we are having, the FDA approved product today. Near about 60% of the product is having the heterocyclic nucleus. So the importance of heterocyclic nucleus we can come to know from this. Near about 60% of the products are having the heterocyclic nucleus, means the aromatic ring having nitrogen, oxygen, sulfur atom. So the Fischer-Indel synthesis under microwave irradiation process in less than 3 minutes at 150 degree Celsius using water as your reflection solvent. But when you are using the other method, you can see 16 hours you have to get. But here just 3 minutes you will get. Here the yield is 62% in conventional method. But when you are using microwave, the yield is 94%. So next one is ultrasound method. So ultrasound we can use it for the synthesis also. So when you are using the ultrasound, the sound will acoustic cavitation, means it will create the bubbles. So when the bubbles will grow and blast, it will produce a lot of heat and energy that will be used for the chemical reaction. Next, continuous flow process. Just now I told this is the conventional batch method. Means we are taking all the reagent together in a single big vessel and do the reaction and get the product at once. But in continuous process, it is a small tube. We are sending the ingredients slowly all together and we are getting the product simultaneously. That is the added advantage. Means here the energy is less and the heat distribution will be 100% proper. So this is the comparison between batch-wise and continuous flow process. So in handling hazardous intermediate, the total continuous flows circumstance, when we are using the continuous flow, the hazardous material will be produced and immediately it will be utilized in small quantities, microbias. The reaction is very slow, means the reactants are the quantity is very less, so the production of the hazardous substance also very less. So immediately it will produce and the next step it will be utilized. So the final product is completely free from the hazardous substance. But in batch wise, it is bulky, so we are getting the bulky product is also having the hazardous materials like all these things. So one of the example is the sildenafil citrate again. So the next one is, instead of normal catalyst, the biocatalyst we can use. Now what is biocatalyst? We can use either amino acid or enzyme or the mutated enzyme. That is called as the directed evolution. That is mutated enzyme we can use for the further synthesis. So next one, just 5 minutes, madam, 2 minutes. So next thing is how we are going to use the metal catalyst like platinum, palladium, ruthenium. If you are using directly, it will be there in the end product. Instead of that, what we have to use? We have to use resin and coat this one with this one. You have to take the resin and interact with your metal. So it will not come out. So, but it will do the catalyst work. But finally, we can easily filter out this one. So the catalyst will not be there in your final product. That is one of the process we can use in green chemistry. Then next one is metal-free organocatalysis. Instead of metallic catalyst, just now you have seen palladium, rhodium and all these things. Instead of that, we can use the organocatalyst, which is containing only carbon, hydrogen, nitrogen and oxygen. For example, we can use this one. For example, we can use this one. The aldol reaction, we can use proline. Proline, you know, it is obtained from the amino acid. We can use that one for the synthesis. Then electrochemistry and photoreduction. So electrochemistry, what is the advantage? When you are using electrode, the oxidation and reduction reaction will take place without using any oxidizing or reducing agent. So no need to add any oxidizing like iodine, potassium permanganate. No need for that, or zinc, all the things. So we can use the electrochemistry and photochemistry. The earlier days we have used the UV radiation. Nowadays we are having the visible radiation to do the reaction. So we can do it with the visible light. So it is a circular economy process. So in circular economy process, we are going to completely stop the wastage. It should be a cycle like you have to design, produce, use and recycle it. There is no space for waste. So you have to design the process like that. So only it is called as circular economy, and we are going to use AI technology here. So already you all are about the artificial intelligence. Here we are going to use the neural technology. Neural technology here like convolutional neural network, graphical neural network, generative neural network. Like that, there are many neural networks are there. By using that, we can design the process. Next one is the retrospective synthesis. What is retrospective synthesis? The normal synthesis, we are mixing A and B and we are going to get the C. In retrospective synthesis, we take the final product, break down into small molecules and see the availability. If it is commercially available, then you can start the synthesis from here. Retro means you have to go back and do the synthesis. Instead of A plus B, C, I am getting C I take, break down into A and B. Whether it is suitable, then I can do the synthesis. So we can get the easiest step to synthesize the product. That is also one of the eco-friendly synthesis method. So whatever I told, we can reduce it. No, we cannot completely prevent the environmental pollution. But we can prevent pollution and maintain the sustainability. That only we can do now, because already we spoiled enough. So there is no chance to completely cure that one. Instead of that, we can try to maintain that for our next generation. So the main aim of sustainability is to make it chemistry easier. Circular means 0% waste, 100% we are going to use the renewable source, recyclable source. So with that, thank you for your attention, and let's build a greener future together for our next generation. Okay, any question?
Thank you, sir, for your valuable talk regarding the sustainability in the pharmaceutical research for green fields. Now the session is open for the students, delegates. If you have any doubt, you can ask the doubt. Are you people are interested to have any doubt? You can clarify that. Fine, the session it is very nice, sir. Actually, I have one doubt actually.
Sir.
You told about that mechanochemical and physical snow. There we are using that solvent state activation technology. We can use lemon for getting that better yield instead of being some other organic compound. Only the thing it is lemon only it is possible. We can use some.
No, you can use any citric acid producing it, any citric acid.
Okay.
Because it is easily available. One is lemon, so we use the lemon.
Yeah, that's what I am
So
thinking.
any citric acid.
Okay.
Any citric acid you can use.
Okay. Actually, you told about that only lemon, no, so that what
Yes, we use the lemon. Yes, it is easily available one. That only the beginning I told you have to use the common sense. So it is easily available. No need to go anywhere. Just you will go and buy and take use it.
Fine.
And cost wise also you have
Yes,
to think.
yes. Fine, it is very nice talk. Actually, sir marked about the particular word about the sustainability. It is a wonderful term. I think how many of you people you might be know about that. The word it is going to be in that another next era. It is going to be wonderful. That word it is most important, sustainability. So sir gave that very good talk about this particular topic. Thank you very much, sir.
Thank you, sir. I request Chairperson Dr. G. Muthu Bhubadi, sir, to honor the eminent speaker, Dr. V. Ravichandran, sir. Now I request Dr. M. Chitra ma'am to honor the chairperson, Dr. G. Muthu Bhubadi, sir. Dear students and delegates, tea and snacks are arranged in second floor. Please kindly be back in 15 minutes for the next session. Thank you.
and 16 books and book chapters. She is actively involved in several funded research projects and has guided many postgraduate students. She has received several prestigious awards, including the Young Pharmacy Teacher Award, and Young Scientist Award, and Research Excellency Award. In recognition of her valuable contribution to pharmaceutical education and research, we are delighted to have her with us today. I now request Dr. Gomati Swaminathan to kindly deliver her resource session. Welcome, Dr. Gomati.
Thank you, Dr. Ruby Madam, for a nice introduction. Thank you so much. First of all, a very good afternoon to one and all. Students, those are standing outside, please come inside. And only, I think maybe my session, only 25 to 30 minutes only. Okay, please come inside and please attend the session. Slide change. Okay, keyboard. Okay, no problem. Those are sitting back, please come front at least. Am I audible? Okay, we'll start. A few people are sitting in classroom. I am so happy about that. Good afternoon, one and all. அனைவருக்கும் மாலை வணக்கம். Okay, all are happy? Okay, now am I audible? Okay, this is an interactive session. You don't think simply I will talk, just you will listen, otherwise you will do the chit chat and all. In between I will come, okay, inside, and I am going to interact with you, okay. So usually I am so happy today and so emotional because I am standing in Vattamalai. Vattamalai means Kumarapalayam. This is very special place, okay. This is very close to my heart. This place is very close to my heart. You don't think, okay, this is the normal place like that, okay. So I feel always the God is, I am seeing the God through the students, through my teacher, through some places, that and all, okay. So today, what is the title? Title also very special title, that is the greener pathway to better medicines. Why the greenery is very special for us? Why? Anyone? Why it is special? Tell something. Not.
Nature of God, ma'am.
Yes, very good. That is the nature of God. My title also you have to look at that, digital herbal intelligence, empowering green drug discovery through natural product databases. Here the very important point is natural product database. databases. So nowadays no need to worry about the research because we all are in stress. All are using mobile phone. We are addicted with all the social media. What other people will think, we are concentrating on others. We are concentrating on society, but we are not concentrating on us. So that's why we want to take care of ourselves. No need, don't be stress, depression and all. Be happy. Whatever you are doing, you have to be happy with that. Okay, whatever you have, so from that you have to எது இருக்கோ, ஓகே உங்ககிட்ட என்ன இருக்கோ அதை வச்சு சந்தோஷப்படுற விஷயமே இயற்கையோடு சேர்ந்தது தான். வாட் எவர் nowadays we are doing that and all, what is that? That is the artificial. செயற்கையை கொண்டு வந்து இயற்கைக்குள்ளே புகுத்துறப்போ நிறைய இஷ்யூஸ் வரும். ஸோ தட்ஸ் வை வி வாண்ட் டு அவாய்ட் இட். நானும் இதே மாதிரி இந்த ஊரில் படித்த ஒரு ஸ்டூடெண்ட் தான், சரியா? ஸோ அதனால் உங்கள் எல்லோரும் கூட எனக்கு எப்போவுமே கனெக்டிவிட்டி வந்து ஜாஸ்தி தான், சரியா? எமோஷ்னலாக. ஸோ வாட் இஸ் த மீனிங் ஆஃப் ஹெல்த்? வாட் இஸ் த மீனிங் ஆஃப் ஹெல்த்? Physical, mental and social well-being. Okay, if you are physically fit or not, physical well-being is very much important. So sometimes we will not get it also, maybe some problem also, not a problem. Physically fit or not, mentally we want to be happy, then only socially we become very good human being. Okay, so that's why so don't feel panic and all, don't talk each other, just listen for 20 minutes, that's it. Okay, so daily people are talking about, scientific community talking about molecular docking. What is the meaning of docking? So first we want to know about that, then we will read about we will talk about the herbal databases. The molecular docking means that they are saying about in silico methods. You have to, you talk to your teachers. Otherwise, what paper they are producing, what is the presentations, what they are doing. In chemistry department, analysis department, they are doing something that is called as docking. So what is the meaning of docking? Means this is an in silico method, computer based process which will predict how the small ligand molecule. What is ligand? Anyone knows ligand? What is the ligand? Small molecule. Any others? Drugs.
Small molecule or drug molecule.
Okay, small molecule, small drugs and all we are saying that is the ligand. What is the meaning of receptor? Proteins. Where it is located? Within us. Now we are going to read about us, our own mechanism, our own traditional medicine. These and all now we are going to read it. Okay, so here you have to look at that. That is the, here we are going to predict the how the small molecule ligand fits into the receptor, how it will fits into our body. Receptor means, I hope you studied in biochemistry second sem. Okay, what you studied? Enzymes you studied, proteins we studied. All the hormones are made up of proteins, all the enzymes are proteins. So we know about that, how it will fits into the receptor binding site, simulating drug target interaction. Here the target means that is also receptor only. Receptor means enzymes which will be located, which is located in our own body only. So ligand they are saying, no, what is the ligand? I think maybe in 4th year B Pharm you are going to do some project, na? So you are going to prepare some drugs, okay, molecules, that is only called as ligand. So you don't think ligand means that is totally different, receptor means totally different like that. You don't think like that. We are going to study about our own laboratory, what I am going to prepare, what I am going to isolate. That molecule is called as ligand. And one more thing, we are going to study about our own body mechanism. So that interaction between the what you are preparing in the lab and and what is in your body, that interaction only we are going to going to talk now today. Okay, so that's why no need to think anything difficult, that and all, don't think like that. Okay, so why the molecular docking is very much important? Previously, I think maybe if I want to one drug, I want to find it, develop one drug means I need nearly 20 years, otherwise 30 years. You have to see the scenario of benzene and all. It was started from the 18th century. It was completed in the 19th century. So like that, no need to waste our time. Okay, that is very much important. Time is very precious. That's why the time is very much important for us. And next one is the cost. Okay, our India is not having only, listen, our India is not having only rich people. Okay, we want to concentrate on moderate people. So economy is very much important. Cost of the tablet is very much important. What you are doing, that time is very much important. The chemical steps also very much important. Very short period, we want to find the good molecule with the less cost. So that's why this is also, that's why now we want to think about the cost and time. And second one is, nowadays computer is available in all the laboratory. Correct or not? Computer is the, so no need to write the mechanism, write the synthesis as like previous days, olden days. Okay, when we was studied, otherwise our seniors was studied, that time they used to prepare the scheme. That scheme also nearly 50 steps, otherwise 15 steps, 15 to 20 steps they used to do. Nowadays no need to do these and all. With the computer we can able to do the drug designing. Okay, because more than us, more than our generation, your generation, okay, you are handling lot of mobile phones and all. So that's why you can easily do the drug discovery with the less cost and less time. So that's why this is very easy, simple process only. And here what we are going to do, it means we are going to identify the potential lead compounds. Here we are not going to write 50 compounds, 100 compounds, then sometimes we will buy all they used to buy all the chemicals, that and all, no need. Just through computer, you have to find one molecule, two leads you have to take it. From that you have to prepare 4 to 6 derivatives. That is more than enough for our research. So that's why don't think research means it is big process and all. Nowadays you have to think all wherever you go, artificial intelligence is the boss of us. Now we have the theme also, that is the pharma AIist. Okay, so in pharmacy profession also we need AI enabled classroom, we need AI enabled everything. So that's why it is easy only. And understand, we want to understand. How we will understand? First you have to read about the ligand. What is your drug? So thoroughly you have to do the literature. What is your target? So that is also you have to focus on it. In target also, so many bioinformatic tools are available nowadays. From that you have to design the hypothesis. Okay, you have to give some proposal. Okay, that proposal should be very novel. Like that we can able to design it. And next one is, we have to predict the binding energy, binding affinity, how the ligand will go to the receptor, how it will bind. These and usually they are doing in this molecular docking. And lead also we can able to optimize it. Okay, so these and all you have to note it. The molecular docking helps the scientist to find the best fitting drug molecule for a target protein. Okay, quickly. So this is very much important. Efficiently for the drug development. So I think maybe now you know that. You don't think more beyond the thinking and all. Just we are taking one ligand. That ligand I am going to prepare in my own lab, where you are studying, where you are working. Then your target is, which was located in our body. Okay, so what is the binding affinity? According to that you have to do with the computer. We are going to do the molecular docking. From that we are going to select some lead. From that lead we are preparing some derivative. Then you have to do the in vitro studies and the in vivo studies. What is in vitro studies? cytotoxic studies. Okay, what is in vitro? From cell line in microbiology department and biotechnology department they are doing na, so that is in vitro studies. Then you have to move to the animal studies, then clinical trials. That's it. This is the small introduction about the So here I am saying molecular docking and all. Here two critical elements are very much important. One is the protein. I think you know that is the receptor. Another one is the ligand molecule. That's it. And proteins are very much important. Already I told you they are only enzymes. They are only doing the transport. Okay, and next they are only providing the structure of our body. Okay, our body is made up of proteins. Okay, so then they are only giving the signaling and they have the immune defense activity. It will act against the any other foreign matters, foreign substances. So that is also you have to note it. So these all the functions are regulated by protein. That's why we want to read about the protein. So you have to read biochemistry well, then HAP also read it well, chemistry read it, then you have to apply in the pharmacology and medicinal chemistry. So don't think you are studying normal subject like that. You don't think. Really you are lucky people, that's why you are in pharmacy profession. Okay, so protein is very very important for us here. And next one is the ligand also I told you. Nowadays lot of bioinformatics tools are available. No need any guide. Okay, guide means just they will guide you. You have to guide yourself. Okay, you have to guide yourself. Lot of bioinformatic tools are available. First you have to select the disease. In which area you want to do the research. That is very much important for us. In genetic level also, these databases will guide us. Okay. So first, for example, my area of research is neurodegenerative diseases. That is the common name. Okay. In that, you have to go narrow, we want to broadly you have to read it. You have to select the area of research. Then you have to narrow it. What is your target? My area of research is Alzheimer's disease. In that, my area of receptor is P2X7 receptor. As like that, in 4th year, otherwise M.Pharm you are going to do some project, no? You have to select some area of research or target. So through this, that and all NCBI gene, Uniprot, Plast, Shrink, KEGG, these and all, I given the link also. I will circulate the PowerPoint presentation. Really, if you need, you have to take these and all. These tools will explain as more than humans. Okay, so just you have to take the area of research, then thoroughly you have to collect the literature, read it, then you have to frame the which receptor you want to do it, which enzyme you want to do the research. Like that you have to select it from the bioinformatic tools. So this is nowadays a lot of people they are aware about the bioinformatic tools. Okay, so and next one is I told you ligand means you know that. Here we are going to read about the ligand and that receptor interaction. These are the types of ligands. The size of the ligand also very much important. It will give more sense to the already sir told no some chemical sense and all. As like that it will give some research sense to the researchers. Okay, so that's why that penetration also very much important. That's why all the studies you have to thoroughly you have to do it, then you have to start the research. Okay, and these are the ligand bases are available. These all are free databases. Whatever you want, we can able to get it. So the ligand databases provide the chemical structures and bioactivity informations and all the things we will get it from through these all the databases. I hope already you knows about the PubChem, Chempel and all. I think most of the people are using the drug bank and PubChem and all. And nowadays these scholars, PhD scholars and all, they are using the coconut databases, zinc databases and all. So from that you have to go and visit the database website and collect the information. No need to draw in ChemDraw and all. That is also essential. So here it will give lakhs and lakhs of chemical structures for drug designing. So that's why taking 10 compounds and doing the research is not sufficient nowadays with computer. You have to take lakhs and lakhs number of the chemical ligands. You have to collect it from the natural databases. Then you have to do the molecular docking. Then you have to select few compounds for the end results. Okay, that is sufficient for the in vitro and in vivo studies. Okay, next. Here, this is only the first slide. Now I given small introduction about the drug design. Okay, so here my title is Digital Herbal Intelligence. Okay, so here plants. Already I think maybe one of the students she told the plant is mother of medicine. What is that? Mother of medicine. This is interactive session. You interact it, then it will be very nice. Okay, so mother of medicine. This is the best gift from the God. This is the best gift from the God. Holistic therapy. Why holistic therapy? How to say? We are believing okay, our own system, traditional system. That's why it is called as holistic therapy, which is integrating the emotional. Emotional is very much important. Then mental and spiritual levels. Okay, so that's why the herbs is very much important. This is only the mother of medicines. Okay, simply we are not seeing the plant as like plants. Correct or not? Okay, everywhere plant is there. While you see this side, you have to see some plants are there. Just to go down, we are seeing that is nature. Okay, this is very much important for us. It will heal the body and calm the mind and uplifting the spirit. So that's why this is the gift from God for a healthier and happier and harmonious life. So this is very much important. The plants are very much important for the research. So why this is having any magic? This plant is having magic. Yes, it is having some magic. So that is only called as phytochemicals, active constituents, phytoconstituents, like that. We used to say, we are seeing simply plant is plant. The plant is having the lot of chemicals. Whatever you are buying that chemical substances, synthetic chemicals, when compared to that, this is low cost. If you have the field, you can able to cultivate it. Correct or not? If maybe it is not sufficient, no need to wait for that chemical company and all. Just you have to cultivate it and collect it. And next one is the second one is always we are believing in the Ayurveda. Okay, Ayurveda, I hope you all know that we are believing the natural plants and herbs. Okay, so that's why this is having the phytochemicals like alkaloids, flavonoids. Lot of phytochemicals are available. Lot of chemical substances are available from the plant. So it is having lot of activity also. So that's why always don't concentrate on that synthetic route and synthetic pathway. Always concentrate on the herbal medications, herbal phytoconstituents. You have to select it for your research. Because why? This is the mother of medicine like that we are saying. Why? All our mothers, I hope here I think maybe how many members we are sitting. Our mother and grandmother is doctor. How? Without MBBS degree. Why? Because if something is happening, immediately they have some remedies. Correct? So that's why you have to believe your mother and grandmother, because they are using the herbs as the medicines without any MBBS. Otherwise, without prescription and all, anything immediately I feel somewhat floating. Immediately they will give some ginger kasayam and all. Okay, correct or not? கீழே விழுந்துட்டீங்க என்ன பண்ணுவாங்க? இமிடியேட்டா மஞ்சள் அரைச்சு என்ன பண்ணுவாங்க? பத்து போடுவாங்க. சரியா? கேஸ்ட்ரிக் ட்ரபிளாக இருந்தால் என்ன பண்ணுவாங்க? இமீடியேட்டா ஜீரக வாட்டர் எடுத்துக்கோன்னு சொல்லுவாங்க, ஓம வாட்டர் எடுத்துக்கோன்னு சொல்லுவாங்க. ஸோ அப்போ வீட்டில் யாருமே படிக்கல. அவங்க யாரும் எங்கள் அம்மாலாம் இப்போ எடுத்துட்டுன்னா ஒரு சாதாரண குக்கிராமத்தில் தான் பிறந்திருக்காங்க. அவங்களுக்கு இதை பற்றி எந்த நாலேஜ் இருக்காது. நான் சொல்கிறேன் ஃபைட்டோ கான்ஸ்டியூன்ட் இந்த ஹெர்பு வந்து மேஜிக் இருக்குன்னு என் அம்மாவுக்கு தெரியுமா, என் பாட்டிக்கு தெரியுமா? அவங்களுக்குலாம் தெரியாது. என் அம்மா எப்படியோ அதே மாதிரி தான் உங்கள் அம்மா, என் பாட்டி எப்படியோ அதே மாதிரி தான் உங்கள் பாட்டி. உங்களுக்கு ஏதாவது ஒரு சின்ன பிரச்சனைனா அம்மா பாட்டிகிட்ட கூட்டிகிட்டு போயிடுவாங்க. பாட்டி வந்து அவங்க ஒரு கை வைத்தியம் வச்சுருப்பாங்க, அதுதான் ஹெர்பல் மெடிசின்ஸு. அந்த கை வைத்தியத்தை தான் நம்ம இப்போ எலாபரேட் பண்ணி சயின்டிஃபிக் வேர்டில் சொல்கிறோம். இந்த மெடிகேஷன்ஸ்லாம் யார் யூஸ் பண்ணது? ஏன் தாத்தா, உங்கள் தாத்தா, நம்ம பாட்டி எல்லாரும் யூஸ் பண்ண விஷயந்தான் இப்போது நேச்சுரல் மெடிசின்ஸ், நேச்சுரல் இன்டெலிஜென்ஸ் அப்படின்னு சொல்கிறோம். தட்ஸ் இட். இது நீ எடுத்துட்டேன்னா வீட்டில் அஞ்சரைப்பட்டி, எத்தனை பேர் வீட்டில் அஞ்சரைப்பட்டி இருக்குது? ரைஸ் யுவர் ஹேண்ட்ஸ். ஸ்பைஸ் பாக்ஸஸ் ப்ளீஸ். நம்ம எல்லாருமே எந்த மெடிகேஷன்ஸை ஃபாலோ பண்ணுறோம்? ஹெர்பல் மெடிகேஷன்ஸ் தான் ஃபாலோ பண்ணுறோம். ஸோ பிகாஸ் வி ஹாவ் சம் அஞ்சரை பெட்டி, ஐ திங்க் மேபி தோஸ் யூ டோன்ட் நோ, யூ ஹாவ் டு see the picture of the அஞ்சரை பெட்டி. மந்த்லி grocery will come means immediately what these people will do. அதுலேருந்து ஜீரகம், மிளகு, ஃபெனல், அதாவது பெருஞ்சீரகம், அப்புறம் வேறு என்ன எடுப்பாங்க? வேறு என்னென்ன தெரியும் உங்களுக்கு? என்ன தெரியும் இப்போ? என்னென்ன தெரியும்? அஞ்சரை பெட்டியில் என்னென்ன வைப்பாங்க? மிளகு வைப்பாங்க, ஜீரகம் வைப்பாங்க, ஃபெனுக்ரேக்கா அப்படின்னா என்ன? அது பேர் என்ன? வெந்தயம், சரியா? சோம்பு. கூலிங் எஃபெக்ட்னா இமீடியட்டாக வெந்தயத்தை எடுத்து ஊற போடுமான்னு சொல்லுவாங்க. சம்டைம்ஸ் பல்ல வலிக்குதுன்னு சொன்னால் இமீடியட்டாக கிராம்பை எடுத்துக்கன்னு சொல்லுவாங்க. கரெக்டாக இல்லையா? கிராம்பை எடுத்து வைமா அப்படின்னு சொல்லுவாங்க. அதேமாரி நம்ம நடந்து போகிற அந்த தடத்துலலாம் பார்த்தா நிறைய செடி கொடியெல்லாம் இருக்கும். சில நேரம் ஆடாதோட எடுத்து ஒரு கஷாயம் வைப்பாங்க. சில நேரம் வந்து நடக்க முடியல, மூட்டு வலி இருக்குன்னா அதுக்கு ஒரு கஷாயம் இருக்கும். ஸோ இது மாதிரி யாருமே எம்பிபிஎஸ் டிகிரியும் படிக்கல, யாருமே பள்ளிக்கூடத்துக்குலாம் போகலை, நம்மளை மாதிரி காலேஜுக்கும் வரல. ஆனால் அவங்க எதை பிலீவ் பண்ணாங்க? நேச்சரை வந்து பிலீவ் பண்ணாங்க. புரியுதா? நேச்சர், இட் இஸ் நாட், நேச்சர் மீன்ஸ் யூ டோன்ட் திங்க் டிஃப்ரெண்ட்லி, தட் இஸ் ஒன்லி த காட். ஓகே, காட் கடவுள்னு சொல்கிறது அந்த நேச்சரில் தான் இருக்குது. ஸோ திஸ் இஸ் வெரி மச் இம்பார்ட்டன்ட் பிகாஸ் மை மதர் யூஸ்ட், யுவர் மதர் யூஸ்ட், மை கிராண்ட் மதர் யூஸ்ட். தட்ஸ் வை வி ஹாவ் லாட் ஆஃப் சொல்யூஷன் ஃபார் மெனி டிசீசஸ். ஸோ நீ இதை பார்க்கணும், வீட்டில் தெரிஞ்சுக்கணும், அம்மா என்ன பண்ணுறாங்கன்னு தெரிஞ்சுக்கணும். கண்டிப்பாக பாட்டி என்ன பண்ணுறாங்கன்னு தெரிஞ்சுக்கணும். பாட்டி கை வைத்தியம்னு சொல்லுவாங்க. ஊருக்கு ஊர் ஒரு மருத்துவச்சி இருப்பாங்க. அந்த காலத்துலலாம் இப்போ மாதிரி ஹாஸ்பிட்டலில் போய் பிரசவம் பார்க்க மாட்டாங்க, வீட்டிலே தான் பிரசவம் பார்ப்பாங்க. ஸோ இதெல்லாம் என்ன? நம்பிக்கை. நம்மளோட நம்பிக்கை எப்படி இருந்துச்சு? ஸ்ட்ராங்காக இருந்துச்சு. அதனால தான் நம்ம நல்ல ஒரு ஜென்ரேஷன், நல்ல தாய் தகப்பன் இருந்ததுனால தான் நம்ம ஒரு நல்ல நிலைமையில இருக்கோம். நீங்களும் நல்ல நிலைமைக்கு வருவீங்க. ஸோ தட்ஸ் வை யூ ஹாவ் டு ஃபாலோ அவர் ஓன் கிச்சன். திஸ் இஸ் ஒன்லி அவர் ஃபர்ஸ்ட் ஃபார்மசி. த கிச்சன் இஸ், யூ ஹாவ் டு ஸ்டார்ட் யுவர் ப்ரொஃபஷன் ஃப்ரம் யுவர் kitchen. Okay, so this is you have to note it. So that is only the first pharmacy. Nature is our best doctor. So this is also you have to believe it. Okay, and natural products in drug discovery, we want to read it here. So you have to take the what you are taking, what your mother and grandmother given, that is the extract only they given. Just they used to take some amount of the leaf, otherwise any bark, otherwise just they used to heat it and they used to give, that is only the kashayam extract, like that we used to say. Nowadays we are focusing on that extract, what phytoconstituents are there, from that how we have to get it, means these and all we will get it from the literature. Then you have to do the extraction and identification also you have to do it, but the way of extraction when I was studied, that is different. Nowadays it is different because no need to do now extraction also. Why? Because that is also waste of time. Just we have lot of chemicals are available which is already derived from the natural sources. That's why this is called as library. Chemical libraries are available. Now I need the piperine, means no need to do the isolation. If I need quinine means no need to do the isolation. Just you have to get it from the company. Then you have to change the chemical, what you have to design it semi-synthetic pathway, otherwise you have to make your own pathway and you have to do the in vitro and in vivo. Why we want to do the extraction and all? Already commercially it is available. Piperine is available and all the flavonoids are, hesperidine is available, example. So just collect it and you have to do the some semi-synthetic modification, alter it. What is the basic nucleus? Just to do the alteration and you have to give the input to the what computer, then finally we'll get the result, we will get the lead. From that lead you have to start your research. This is very simple method, no need to do the extraction and all. We are starting from here, we are collecting these chemical constituents and we are going for the in vitro and in vivo and all. And the herbal also we know that this is having the rich chemical diversities. Just you have to do some extract. Next day you go for the phytochemical studies, means we will get lot of chemicals. So that's why you have to choose the natural medications for your research. And this is having the proven therapeutic potential and better and safer tolerability, cost effective, and these and all. We want to avoid the chemical pollution for the environment also. That's why you go with the natural product research. So these and all challenges also there. You don't think simply we do the natural research, we have lot of problem. So we want to that challenges and all we want to convert into the what? That is the good results. So you have to take care of each and everything. So these are the challenges we want to cross it. And here next one is the why nowadays very much important digital herbal intelligence. Because we all know about the turmeric story, you know that na? Turmeric story, you know that na? நம்மளோட மஞ்சள் வந்து இன்னொருத்தர் வந்து பேட்டன்ட் வாங்கியிருப்பாங்க. ஸோ அந்த மாதிரி நம்மளோட நெக்ஸ்ட் ஜென்ரேஷன்ஸ் யாரும் இருக்கக்கூடாது, நம்ம குழந்தைங்க, நம்ம பேர குழந்தைங்க. அப்போ ஸோ யூ ஹாவ் டு டேட்டா கலெக்ஷன்லையும் டேட்டாவை சேவ் பண்ணுறதுலையும் இந்தியன்ஸ் ஆர் வெரி புவர். ஸோ தட்ஸ் வை வி வாண்ட் டு ஒரு சின்ன கெமிக்கல் டெஸ்ட் நீ உன் லேபில் பண்ணாலும் அதை எழுதி பழகு, எழுதி வச்சு செய்யுங்க. அதை ஒரு டிஎல்சி பண்ணுறீங்களா, சால்வெண்ட் சிஸ்டம் செலக்ட் பண்ணுறீங்கன்னா அப்போ நீங்கள் எழுதுங்க. என்ன நீங்கள் பிஃபார்ம் படித்தாலும் சரி, எம்ஃபார்ம் படித்தாலும் சரி, எந்த ஏரியா ஆஃப் ரிசர்ச்சாக கூட இருக்குது. ஒரு ரிசர்ச்சுக்கு நீங்கள் போயிட்டீங்கன்னா ஃபஸ்ட்டு வி நீட் ஒரு நோட் புக் கண்டிப்பாக வேணும், கண்டிப்பாக அதை எழுதணும். எழுதுகிறப்ப தான் தென் யூ ஹாவ் டு எவ்ரிதிங் யூ ஹாவ் டு தென் இன்புட்ஸ் ஹாவ் டு கிவ் இட் டு த கம்ப்யூட்டர். எழுதிட்டீங்கன்னா டேட்டா இருக்கும். டேட்டா இருந்துச்சுன்னா அது பல தலைமுறைகளுக்கு என்ன ஆகும், யூஸ்ஃபுல்லாக இருக்கும். சரியா? ஸோ அதனால் எதுவுமே சும்மா நான் பண்ணேன், ஒரு மேலிஷ் ரீஏஜென்ட் நான் ப்ரிப்பேர் பண்ணேன், நான் சும்மா ஒரு கார்போஹைட்ரேட் அனலைஸ் பண்ணேன்னு விட்டுட்டு போகக்கூடாது. அதுக்கு ஒரு எவிடன்ஸ் வந்து க்ரியேட் பண்ணும். கம்பல்சரி வி வாண்ட் டு க்ரியேட் அவர் எவிடன்சஸ். இப்போ நீ பார்த்தீங்கன்னா ஒரு பேப்பரை அக்செப்ட் பண்ணணும்னா பணம் கொடுத்தும் போடலாம். ஆனால் நீ உண்மையாக பண்ணியிருக்க எக்ஸ்ட்ராக்ஷனை நீ ஃபோட்டோ எடுத்து வச்சுக்கோ, நீ பண்ணியிருக்க ஃபைட்டோகெமிக்கல் ஸ்க்ரீனிங்கை ஃபோட்டோ வீடியோ எடுத்து வச்சுக்கோ. இப்போல்லாம் ஜியோ டேக் இருக்குது, அதை யூஸ் பண்ணி நீங்கள் எடுத்துட்டிங்கன்னா உங்களுக்கு ஒரு பேப்பர், இப்போ ஃபார் எக்ஸாம்பிள் பங்களாதேஷ் ஜேர்னல் ஆஃப் ஃபார்மகாலஜியில் ஒரு ஃபார்மகாலஜி ப்ரொசீஜர் வீடியோ வந்து கம்பல்சரி கேட்பாங்க. அப்போ நீ ஃப்ரீ ஜேர்னலில் வந்து பப்ளிஷ் பண்ணலாம். ஸோ சின்ன விஷயம்னாலும் ப்ராப்பராக பண்ணால் வி வில் கெட் த பெட்டர் ரிசல்ட்ஸ் ஆல்சோ வில் கெட் இட். அது ரொம்ப நல்லா பியூட்டிஃபுல்லாக இருக்கும். அதுக்கடுத்து நீ பார்த்தீங்கன்னா நேராக this is only my main part of this slides, that is the natural product databases are available. This is our own database. What is that? IMPACT, our own Indian medicinal plant databases. Lot of things are available in IMPACT. And next one is collection of open natural product databases, that is the coconut. And next one is the NPAS. Like that all the databases are available, no need to search and no need to do these and all. Just you go and type it, all the details it will give, lot of chemical structures it will give. Then you have to collect it and do the research. Okay. And next one is ethnopharmacology, and next one is the digital documentation is very much important. The traditional wisdom is converted into scientific validation. Finally you have to document it, it will be useful for the future. So this is very much important. And next one is the then nowadays I think maybe AI already incorporated, all the teachers are updated with AI and all. So whatever you want to do, even slides also, just you have to give the prompt, tick tick tick, 2 minutes it will come all the result. No need to waste our energy and all. Within 2 minutes, within half an hour we can able to prepare each and everything. So no need, you don't think number one college only I want to study that and all. Even you are studying in government college, government school, how you are looking at you, how you are transforming you, that is very much important for us, because we all have the mobile phone. Just you have to give the prompt, it will give all the results, it will guide you. Okay, so this is very much important. And next one is I given, so just you have to click it also. Here if you click means directly it will go to the databases. Just every databases you have to check it. For example, Indian medicinal plants, phytochemistry and therapeutics 2.0. Here how many books are available? More than 100 books are available, and nearly 7,000 published articles are available, and a lot of phytochemicals are available. You can able to see the phytochemicals, and even we can able to check the therapeutic uses also. Don't write it. Cucumber will treat the neurodegenerative diseases. Don't fear for negative results. Give the positive results. Correctly you have to analyze it, then you have to do it. Otherwise it's don't give the fake results for the future generation. Okay. So like that all the databases are available. Please, they have login also, sign up also. You have to do it. You'll get all the things. Okay, coconut databases, zinc databases, PubChem also we can able to collect it. MPAS also. Okay, here also. So you have to see how many 2,04,023 natural products are available. How many targets also you have to look at that. 8,764. You have to see the activity records and composition records and all. You have to look at that. Okay, so no need. As like our Indian, many countries they have their own databases. This is the example of traditional Chinese medicine integrated databases. Also you have to see lot of informations are available in these databases. Okay, this is also you have to note it. So natural molecules are really it will make some sense and the impact only. That is the asset of our nation. Okay, so that's why very carefully we want to deal with that. And green discovery pipeline is you have to look at that. First you have to thoroughly study about the natural databases, then you do the extraction. Otherwise you have to buy the chemical which is belongs to the water, that is the relevant to the nature, and then you do the bioactivity screening, and if you want to do the isolation and all, you do it. Otherwise just you have to collect it, semi-synthetic pathway you have to make it and synthesize it, then do the spectral characterization, then you have to follow the in vitro, in vivo and clinical studies and all, you have to do it. And the future trend is shaping the next era of drug discovery with digital twin, that is also very much important, and generative AI and quantum computing and all. And lot of challenges are available. Okay, complex. It's very difficult to isolate. You don't think this herbal, what these people are doing in herbal research, they are doing easily like that. Simply they will take plant. Or plant எடுத்தா ஏதோ ஒரு research முடிச்சா அப்படி நினைக்க கூடாது. அதில் நிறையா ப்ராப்ளம் இருக்குது. சிந்தட்டிக் கெமிஸ்ட்ரி கூட ஈஸியாக இருக்குது. ஒரு சிந்தசிஸ் ஃப்ரேம் பண்ணிவிட்டு நீங்கள் சிந்தசைஸ் பண்ணால் கண்டிப்பாக ரிசல்ட் வரும். கண்டிப்பாக ஏதாவது கொஞ்சம் சால்வெண்ட்டை மாற்றினா ஏதாவது ரிசல்ட் வரும். ஈஸியாக ஐடென்டிஃபிகேஷன் பண்ணலாம். ஆல்ரெடி உங்கள் மைண்டில் ஒரு கெமிக்கல் இருக்கும், ஆனால் அந்த நேச்சருக்கு எந்த ஐடியாவுமே இருக்காது. நம்ம ஓன் இன்ட்ரெஸ்ட்டில் தான் இதை நல்ல ஒரு இதுக்கு கொண்டு வர முடியும். ஸோ லாட் ஆஃப் ஆப்பர்ச்சுனிட்டிஸ் ஆல்சோ அவைலபிள். நவடேஸ் சார் டோல்ட் ஒன் பாயிண்ட், தட் இஸ் த sustainable development goal, that is very much important. That 3 they are comparing, that goal 3 is a good health, like that all the parameters are very much important. So that's why we have to think about that SDG also. So lot of opportunities also available. In conclusion, just you have to look at your back, that's it. Back to nature, forward to a better future. Always you have to believe your own system, traditional system. That is very much important message. Let us reconnect with nature and choose herbal wisdom for a healthier, safer and greener tomorrow. And next one is, this is our team. Okay, our team is constitute of 7 staffs, Dr. R. Kalirajan, Dr. Gauramma, Dr. Jubi, Dr. Gomadi Sanish and me and Dr. Durayanand and Dr. Srikant Jupudi. All are doing very good research. Our team is doing very good research, especially our objective is hit to lead optimization, innovative process development for synthesis, drug and drug intermediate, API handling, patent and regulatory affairs. Especially we are doing the research on the cancer and neuroprotective research. 2 Gomadi madams are doing neuroprotective research. Dr. Srikant is doing the cancer and anti-tuberculosis. Remaining all the 3 teachers are doing cancer research only. If you need any help, please contact us. We have one center also, Center of Excellence. We are doing the consultancy service also. If you are not able to do your research, please contact us. Fast 30 minutes I am talking about nature and all. I don't know about when I was studied B.Pharm in RVS, I don't know about research. I went for group project and all. Okay. When I studied here in Vattamalai, that time I went to that natural product research. Whatever the things my teacher taught, no, that is only made me very strong woman. Especially I received nearly 4 to 5 funding. One is from ICMR, that is nearly 24.72 lakhs, and next 50,000 seed money grant from JSSHR, and 3 lakhs from JSSHR, and 5 lakhs for center also we received. That all the researchers which belongs to natural product research. You have to see the title also, everything which is relevant to the natural. So you don't think I studied in this college, that college and all. If you believe, I am very sure you will get it something special. That's it. Always you will get some miracle also. So all the things are natural product research only. You have to see that documentation I am saying, no, the documentation is very much important. Guide means you don't guide it. You are also one of the part of your research. So do it, take the photo, then you have to see the memories. I think maybe after 40 years it will be the very nice experience, and we are creating our own story. So that's why these and all very much important for us. Don't focus always on that research, just you have to see the community. We started to do some outreach program. All the lady staffs we used to do the outreach program for, especially for tribal group and orphanage kids. Here maybe tribal is not available. Lot of orphanages are there. Focus on that orphanage and all. Write small seminar grant for CSIR, ANRF and all the grant division, national grant division. You have to write it. I am very sure you will get it. What is your area of research? What is your focus? That is something different, means you will get magic. That's it. So these are the outreach program glimpses, and this is published in all the articles and all. So you have to file it. Documentation is very much important. Like that you have to do the documentation. Choosing happiness is a decision to trust your heart over your analytical mind. You have to choose your area of research. What is your interest? What your soul will like? That is only very much important for as a human, for a human life. So this is very much important. I think maybe very short time, 30 minutes is not sufficient to explain about these all the things. Okay, so thank you so much for I think maybe I thank the management, JKK Muniraja management, for giving opportunity to discuss among the young minds. Why? Abdul Kalam sir always, I think maybe she is also my student, and Guru also my student. So always she told one word, okay, knowledge gives creativity, creativity leads to thinking and all. What is learning is?
Learning gives creativity.
Creativity leads to thinking, thinking leads to knowledge, knowledge makes you great. I feel very proud. Always whenever we used to conduct some program, I used to quote Abdul Kalam words. So same thing only my next generation also, I felt very happy about her. So the creativity and all, where it will be? The best brains of the nation. The best brains of the nation. sitting in the last bench. Okay, so that's why students are our wealth. You are only our ambassador. So read well, feel, make proud to our guide. உங்கள் டீச்சர்ஸை நீங்கள் சந்தோஷப்படுத்தினாலே போதும், அம்மா அப்பாவை சந்தோஷப்படுத்தினாலே ஒரு நல்ல ஒரு ஹாப்பியான. எனக்கு கிடைச்ச டீச்சர்ஸ், எனக்கு கிடைச்ச அம்மா அப்பா மாதிரி எங்கேயுமே கிடைக்காது. அதே மாதிரி தான் உங்கள் எல்லாரோட அப்பா அம்மாவுமே ஸ்பெஷல் தான். ஸோ ஆல்வேஸ் யூ ஹாவ் டு தேங்க் த காட், பேரண்ட்ஸ் அண்ட் டீச்சர்ஸ். தே ஆர் ஒன்லி த பில்லர் ஆஃப், தே ஆர் ஒன்லி த, நமக்கு தெரியாது, நமக்கு கீழே இருந்து பின்னாடி நிறைய பேர், ஒரு எவ்ரி லீடர்ஸ் வில் பி கிரியேட்டட் பை பின்னாடி, நமக்கு பின்னாடி இருப்பாங்க. அவங்களால தான் நம்ம இங்கே நிற்போம். ஸோ அது அவங்கள எப்போவுமே ரெஸ்பெக்டாக நடத்தணும். பேரண்ட்ஸை, டீச்சர்ஸ், கைடெல்லாம், சீனியர்ஸ் எல்லாரையுமே ரெஸ்பெக்டாக நம்ம நடத்தணும். ஸோ ஐ தேங்க் த மேனேஜ்மெண்ட் அண்ட் பிரின்சிபல் சார். ஹீ இஸ் மை அலுமினி ஆஃப், வி ஆல் ஆர் அலுமினி ஆஃப் JKK, மை சீனியர் who are all sitting here. Okay, I thank these all the people and staffs, Venkadachalam sir, Kalai Chelvi madam, Chitra madam, அவங்க கலை மேடம், these all the people are ரெண்டு கலை மேடம், ரெண்டு சித்ரா மேடம், அப்புறம் இவங்களாம் என்னோட சீனியர்ஸ். இவங்களாம் பார்க்குறப்ப ஐ எம் ஸோ ஹாப்பி அபவுட் தட். என் சொந்த ஊருக்கு போனால் எப்படி ஹாப்பியோ, ஈரோடுனா ஒரே ஹாப்பி தான், உடனே கிளம்பிடுவேன். ஊட்டியெல்லாம் எனக்கு ஞாபகத்திலே இருக்காது. இந்த மண் அப்படி, இங்கேருந்து யார் ஒன்றுமே தெரியாமல் வந்தாலும் என்ன மண்ணை பொண்ணாக்குற பூமி இந்த ஈரோடு மாவட்டம். அந்த ஈரோடு மண்ணின் மைந்தன் மாதிரி நானும் ஒரு பெண் தான், ஈரோடு பெண் தான். சொந்த ஊர் தஞ்சாவூராக இருந்தாலும் ஈரோடு தான் எனக்கு வாழ்க்கை கொடுத்துச்சு. வாழ்க்கையில் எப்போவுமே பழசை மறக்கக்கூடாது. ஸோ ஐ தேங்க் ஆல் த சீனியர்ஸ், டீச்சர்ஸ், எவ்ரி ஒன், அண்ட் கோஆர்டினேட்டர்ஸ் ஆஃப் இன்டர்நேஷ்னல் செமினார், ஸ்டூடெண்ட் volunteers, camera, and all the people who are all organizing for all, and delegates especially. You are only the core strength, heart of the seminar. At least you people are sitting here. I am so happy about that. Whether you are listening or not listening, that is not a matter, but you people are here. That's why I feel very proud and happy. That's it. Okay, finally JKK Muniraja, Annai, JKK Sampurani Ammal College of Pharmacy, Kumarapalayam. Thank you so much. Any queries, please contact me. Any queries? Any questions? No questions.
Ma'am, I have a question.
Yes, எங்கே?
Here.
சொல்லுங்க.
Can digital herbal intelligence help to standardize the herbal formulations?
Yes.
Ma'am, how?
How means doing, you have to say, doing lot of things, you have to restriction we have to do it. இப்போ plenty of number of chemicals வச்சுட்டு நீ ஸ்டாண்டர்டைஸ் பண்ணுறதுக்கு போராடுறதுக்கு பதிலாக இந்த டிஜிட்டல் இன்டெலிஜென்ஸ், அதாவது இந்த கம்ப்யூட்டர் எய்டட் ட்ரக் டிசைன் தான் டிஜிட்டல் இன்டெலிஜென்ஸ்னு சொல்கிறோம். அதை எடுத்துகிட்டு இருக்கப்போ என்னென்னா, நீ நம்பர் ஆஃப் கெமிக்கல்ஸ், நிறைய காஸ்ட் தேவையில்லை, நிறைய விஷயம் நம்ம போராட வேண்டாம். கொஞ்சமாக நீ எடுத்துக்கலாம். ஒரு 10 கெமிக்கலாக ஒரு 10 டெரிவேட்டிவ்ஸ் எடுத்துகிட்டு அதை ஸ்டாண்டர்டைஸ் பண்ணுறப்போ வில் கெட் சம் பெட்டர் ரிசல்ட். ஆனால் கம்ப்யூட்டரில் வர்ற எல்லாமே 100% ரிசல்ட் தரணும்னு கிடையாது. பட் இது ஒரு சின்ன ஸ்டெப்பு. என்னென்னா, நிறையா வருஷத்தை குறைக்கலாம், நிறைய எனர்ஜியை குறைக்கலாம், கொஞ்சம் ஸ்மூத்தாக ஒரு ரிசர்ச் பண்ணலாம், அவ்வளோதான். மற்றபடி இது சப்போர்ட் பண்ணும். நம்மளுக்கு சக்ஸஸ்ஃபுல்லாக வந்துருச்சுன்னா சூப்பர் தான். சக்ஸஸ்ஃபுல்லாக என்ன பண்ணணும், திருப்பி அகெயின் யூ ஹாவ் டு டூ ஃப்ரம் த ஃபர்ஸ்ட். அவ்வளோதான், முடிஞ்சது.
Okay, ma'am.
Any other question? That question from my own student. Any other questions? Okay, thank you so much. Thank you.
Thank you, ma'am. Now I request Chairperson Dr. S. Ruby ma'am to honor the eminent speaker Dr. Gomati ma'am. I request Dr. A. Chitra ma'am Associate Professor, Department of Pharmaceutical Chemistry, to honor the chairperson, Dr. S. Ruby ma'am.
The next section about to start. I request Dr. B. Balan sir, Professor and Head of the Department of Pharmaceutical Chemistry, Erode College of Pharmacy, to kindly address today's eminent speaker, Dr. Parasuraman sir.
Good afternoon. Good afternoon. Thank you. Listen, today's the third session is going to start. This session will be very shortly will be completed. Don't worry. Another 40 minutes by maximum of 30 to 40 minutes. Okay. So please listen. Okay. Good morning. Sorry, good afternoon. The third session is going to start now. The third resource person, Dr. Parasuraman P. It's my great privilege to introduce our esteemed resource person, Dr. Parasuraman P., Associate Professor, Department of Pharmaceutical Chemistry, Faculty of Pharmacy, MS Ramaya University, Applied Sciences, Bangalore, Karnataka. Dr. Parasuraman is renowned academician, researcher, pharmaceutical scientist with over a decade of experience in the pharmaceutical education, research and drug discovery. His area of expertise including computer-aided drug design, medicinal chemistry, molecular docking, nanotechnology-based drug delivery, and pharmaceutical research. He has an outstanding research profile with more than 136 publications in Scopus and SCI indexed journals. He published 7 books, 7 patents, and several funding research. He guided numerous UG and PG and doctoral scholars. He delivered several keynotes, lectures, workshops, and invited talks in the national and international level. Dear students, please, silence please. Dr. Parasuraman has received several prestigious awards for Young Scientist Award, Annual Exemplary Faculty Award, and Best Researcher Award. He is the editor and reviewer of many internationally reputed journals by Springer, Nature, Elsewhere, etc. We are truly honored to have such an accomplished researcher inspiring the academician with us today. I extend my warm welcome to Dr. Parasuram P. and invite him deliver his talk. Welcome, sir, please.
Thank you, sir.
Welcome, sir. Another 30 minutes, please, quiet.
Hello. Hi. Sharp 30 minutes. Sir said 40 minutes. No, only 30 minutes with the images. Right. So many, what do we call, right from the chief guest of honor to speakers. They speak about drug and all. First we should know what is drug, no? What do you mean by a drug? What do you mean by a drug? What do you mean by a drug? It's a chemical? Is it a biological? No, it can be anything. So drug means any particle which is intended for the treatment, mitigation, prevention of any disease in man or animal. That is called as drug. And it can be from chemical sources or biological sources or biotechnological sources, maybe synthetic or non-synthetic. And source of drug. So most probably we are getting many drugs from various sources, maybe from the plant sources, animal sources, synthesized biotechnological product. As ma'am have explained more about the plant sources, right? So I don't want to involve in those things. So many are the third year, second year, final year students, right? And some will be masters. So you have studied the classifications, maybe the medicinal chemistry classification, maybe the pharmacology classifications, even though each classification will have 5 to 10 drugs. Yes. Then why we need new drugs? What might be the reasons for getting a new drugs? Why we have to synthesize a new drug? Why we have to find a new drugs? Is there any reason? Yes. The first reason is unmet medical needs. Unmet medical need. What is that means? We couldn't able to meet up the regular medical needs because new diseases are coming, new targets we are identifying. So there is a need for finding a new drugs. Maybe example, previously we had COVID, swine flu, then now monkeypox, Nipah virus. Day by day, new viruses are coming, new diseases are coming. We have to find a new medications. And then the low efficacy. If you think about the dementia, cancer, we don't have a proper treatment. We should find a best drug with more effective in treatment. And then the next is side effects. If you see all the English medicines, with the slide will be there, no? Side effects. All the drugs will have more side effects. And if it is related to hormone, there more amount of side effects will be there. So we have to find a new medications for that. And then the cost of the therapy. If you take some medications, the medications cost will be more. Maybe example, a cancer. Cancer for injections, 5 lakhs, 10 lakhs, where it is not affordable by a lower middle class or upper middle class or anything else. So such a conditions, we have to find a new medicines at low cost to treat the everyone. And then sustained industrial activity, the more important thing is the term called as patent. Mama said, right, the curcumin and those things and all. The patent is more important. All the medical industries are big mafia. That's the right word I can use. Big mafia. Because every month, if you see the stock, sometimes Pfizer will be the top, Reddy's will be the top, Ranbaxy will be the top. Why? Because based on the product selling. So always the company will try to find the medications where they want to be a monopoly. Only monopoly. I am being a monopoly, then only I can sell my product. You don't have a choice. You have to buy that. That's the reason. So company X, maybe company X, they have tried to invest the old failures, current and future. Based on that, they have found a new medical. It's a monopoly, and they'll be selling for 1,000 rupees one strip. Second, after sometimes you'll be getting back. Company B, a company Y, I can say. So they will go, they will get rights. Can I use that molecule? And I'll choose a different formulation method, something else. Okay, fine, you go, you pay me something. For that patent, they will pay. So the company Y will be selling the same medications with a different formula, different combinations for 500 rupees per strip. And after some 6 years or 8 years, the patent will get break. When the patent is break, anyone can synthesize, anyone can do marketing. The drug is called as generic medicines. Have you heard about the branded drug and generic medicines? So the generic medicine in the sense, when the patent is broken, the drug will be marketed by anyone. So what happens, I'll let it be, I'm a local vendor. I'm a local small generic company I'm running. I saw this molecule is potential. I'll get APA from China, because China is the first producer of APAs, medicines. So I'll put a call and I'll be getting, for me the manufacturing cost, one cost, not more than a single tablet 2 rupees. So I know this person, my friend, the pharmacist. Dude, you sell this product, how much? 100 rupees. Company X is 1,000, company Y is 500, and it will be 100. At that time, many might have faced. If you go to a pharmacy and if you ask the medications, do you have this? Same molecules available, different brand. Can I give? Yes, every pharmacist will have the same kind. At that time, what happened? The main company product sale will be reduced. At that time, these people will try to find a new target and new drug discovery. That's the reason many companies will be always try to be a monopoly. The company which is monopoly, which are found on medication, they will be in the top of the stock markets and the what we call the sales and other things and all. And then the drug resistance. Do you know about the AMR, antimicrobial resistance? Yes. What happens? Resistance. Resistance in the sense, சொர்ணம் இல்லாமல் இருக்கிறது. சின்ன வயசில் இருக்கும்போது ஃபஸ்ட் அடிப்பாங்கல்ல, அடித்தா வலிக்கும், அப்புறம் அடிச்சுட்டு போ. We are resisted. So such a way, what happens? If I'm not well, I'll be trying to find as an elder, I'll go to pharmacist. Bro, can you give me medications? I'm not feeling well. The pharmacist may try to explain, yes, you have to take a course of antibiotic. The term course of antibiotic, at least 6 dose, 4 dose, 8 dose, like that. Each medication will have its own thing. What happens? We will think, oh, this pharmacist want to earn money. அவங்க காசு சம்பாரிச்சு நான் ஏன் காசு கொடுக்கணும், 2 செட்டு மட்டும் கொடுப்பா அப்படின்னு வாங்கிட்டு வந்துருமா. ஸோ ஒரு செட்டு போட்டேன் ஓரளவுக்கு ஓகே, 2வது செட் போட்டேன் ஃபார் பெட்டர், அண்ட் 3rd செட் வாடா சண்டைக்கு அப்படின்னு போகிற மாதிரி ரெடி ஆகிடுவேன். எஸ், I'll be ready. But what happens actually in the sense, inside my body, my organism, the microbial organism present, right, almost 70 to 80% will be killed, 20 to 30% will be alive. Where my immune power is more, that's why it cannot able to show its own power against me. So slowly it will be colonizing, multiply. After multiplication, what happens? Again, I am not feeling well. I'll go. Not feeling well. Again, this routine 4 to 5 times in the sense, automatically there is a term called as Darwin's theory. Survival of the fittest. Based on that, what it will do? It will try to change its nature where the drug cannot able to penetrate into the organism. If it is not able to get penetrate into the organism, the drug won't be effective. The disease condition will be more. At that time, we have to find a new medication. It's simple. We may have a mobile with a fingerprint. If I keep, in the same way, if I keep a layer, transparent paper or something, if I keep, it will work. No, it won't identify. It has to travel. That mechanism is called as quorum sensing. It will change its nature so that the drug won't be efficient. At the time, we have to find a new medication. These are the various reasons why a drug discovery is required. So this is the general process. So you know very well, first we have to identify disease, which protein is responsible, what type of drugs I can give, and I should go for a filing a what do we call IND, investigation of new drugs. Investigation of new drugs. Before that, there are the term called as formulation is more important. Why formulation is more important in the sense, what type of disease we are going to treat, what type of people we are going to do. I'll take the National Drug of India. Which is the National Drug of India? Dolo. Paracetamol. Yes, anything paracetamol, like that. So why I am taking a paracetamol in the sense, paracetamol will be available in the form of tablet. Yes, suspensions, syrups. Yes, 3rd, parenteral injections. Yes, why? It is for the treatment of same thing, fever. Let it be antipyretic. As I'm an elder, I can able to swallow to take a tablet, then it's fine to go with the tablet. I'm a person, elder, but I'm unconscious, drug cannot be taken. So I can able to take us through a parenteral. But kid, for a kid, I can't give us a tablet, I can't give us a injection will run. That's why I have to give a suspension. So what type of disease, what type of people we are going to give, based on that the proper formulation should be done. Then I should go for an IND, investigation of new drug, on the humans. On the humans, if I'm going to do a trial, and then after that I'll go for a NDA, new drug approval. Then only a drug will be coming. So this will take a quite long time process. But how technology is going to impact? So, and these are the various problems which will be faced during a drug discovery. If I have a hypothesis, 10,000 molecule can able to work to treat this so and so in the sense, at that time we can able to do. But these are the companies. It should be selective, it should be potent, it should be stable, blah blah blah, like that. Like choosing a mobile and a Flipkart. My camera should be like this, my battery should be like this, my RAM should be like this. The same way here. So how technology is impacting? So we are in the era called as omics era. Omics era, O-M-I-C-S, omics era. Proteomics, genomics, metabolomics, like that, the genomics era, where till the time process which is taken for up to animal studies we can cut down using the help of technology. But mandatory human trials are required. So that space we can able to cut down. So what is CAD? Computational drug design, computational drug design. This is the term. What it will do in the sense, it's two things. One is mathematical formula, another one is computer program. I have a mathematical equation. With the help of computer programs, I'll generate an equation where if I give a chemical structure, it will predict and say this chemical structure may act like a drug or not. Mathematical formula, computer program, if I feed a structure, it should say whether it will work like a drug or not. That's all. That's a computational drug design. Why it is required in the sense? Only two reasons. One is to cut down the time, another is to cut down the cost of the projects. So the term is always whenever you are doing a drug discovery with the help of computers, it should be called as in silico. In vitro, outside the body. In vivo, inside the body. In silico, inside the computers. Why? Because computers are made up of silicon connectivities. The board is made of silicon. That's why the study performed to evaluate, to find a drug through the computer's help in the sense, it will be called as in silico. So this is a general pattern of a drug discovery, CAD. So one is fragment-based drug design, structure-based drug design, ligand-based drug design. Fragment means two things are required total. One is protein structure, another one is ligand structure. If I know only the protein structure, I can go with the fragment-based drug design. If I know protein structure and if I know ligand structure, I can go with the structure-based. If I doesn't have idea about the protein, but based on the structure, then it is called as ligand-based drug design. This is the three methods which we are going to follow or we are going to work out. So one is fragment-based drug design in the sense de novo drug design. If a new disease, example, COVID it came, at the time M-Pro protein they said, where there is no idea which molecule will work out. That time what they'll do is they'll find out the active packet or active cavity. Active packet or cavity in the sense, if you take a protein structure, the protein structure is made up of n number of amino acids. There 3 to 4 will be a key amino acids. Like in your class, a representative will be there, right? If I call and inform, tomorrow class is not there, class is there, exam is there, in the sense, it will spread to all. Same way, the active packet we have to find out and we have to design a drug. Some place we may need a donor, acceptor, or aromatic group, whatever the thing. Based on that, if you sit and keep one on one side, another side, then you will get a drug. This is called as fragment-based drug design. And then the pharmacophore modeling. So in the pharmacology classifications, medicinal chemistry classifications, you will be studying based on chemical structures, coinolol nucleus, pyridine nucleus, pyrazole nucleus, right? But in pharmacology, you will be working based on mechanism of action. For one particular mechanism of action, 4 to 5 drugs will be used, where that 4 to 5 drugs will have different structures, different chemical nucleus. Example, carbonic anhydrase if you take, one compound will be on one nucleus, another nucleus, like that it will be of multiple compounds, but it will be producing activity. So there we have to find out what are the common features, where it is present. That is called as pharmacophore modeling. Pharmacophore, oxophore. Pharmacophore in the sense responsible for activity. If I superimpose those structures, I'll try to find out which is responsible, which position is responsible, where a donor should be present, where an acceptor should be present. This is called as pharmacophore modeling. Then QSCR. So you know about the SCR, right? Structure-activity relationship. When it's a 1D. If in this position, electron donor should be present, electron acceptor should be present, whether the activity will increase or decrease. Same way, if you calculate the quantity, different properties will be there. So electronic property, topoisomerase property, like that, n number of properties will be there. In that, which property is responsible for biological activity? If you are predicting, then it is called as QSAR, quantitative structural activity relationship. So virtual screening. So you know about the high throughput screening general? No. So maybe my friend who is in the synthetic chemistry, he is synthesizing 1,000, 2,000 compound, 3,000 compound. Just synthesize, purify, store it, characterize and store it.
he doesn't have idea where it will work out. Another friend from biotechnology or a pathophysiology identify this enzyme I need to kill, find in the sense. Randomly, we will take all the 10,000 compounds and we'll do an assay and we'll predict, yes, for this particular enzyme inhibition, I need so and so nucleus. That nucleus can able to work out. That is called as high throughput screening. Same thing if you perform using a computer, that is called as virtual screening. Virtual, email, EHTS, electronic, we are saying no, email, the same E, that one will be used. I think molecular docking ma'am has said, but even though I'll just give a simple. Molecular docking in the sense, how effectively a drug or a chemical binds to the receptor. How effectively a drug or chemical binds to the receptor. Why it is required. So do you ever think, I may have a fight with this fellow. I was taking so much time and he wanted to throw me. I just fall down. My hand got inflamed. கை வீங்கி போச்சு. நான் இப்போ ஒரு மாத்திரை எடுத்தாகணும், NSAID, நான் ஸ்டீராய்டல் ஆன்டி இன்ஃப்ளமேட்டரி. ஸோ இந்த கை மெக்கானிசம் என்ன ஆகும், என்ன மெக்கானிசம்லாம் இன்க்ரீஸ் ஆகும்? அரக்கிடோனிக் ஆசிட் பாத்வே, ப்ரோஸ்டோக்ளாண்டின்ஸ், எஸ் சம்வேர், COX-2, COX-2, எஸ் COX-1, COX-2 படிச்சிருக்கீங்களே. ஸோ அந்த COX-2 எடுப்பேன், செலிகாக்சிப், வெலிகாக்சிப், ருஃபிகாக்சிப்னு. அப்போ எடுத்தோன்னே என்ன ஆகுனா, இட் ஹேஸ் டு கோ ரீச் பைண்ட் வித் தி COX-2, suppress the action of COX-2. Underline the word bind with the COX-2 and suppress. ஸோ பைண்டிங் இங்கே தான் இம்பார்ட்டன்ட். அப்போ அதையே வந்து நம்ம கம்ப்யூட்டரில் பார்த்தோன்னா அது பேர் தான் மாலிகுலர் டாக்கிங். ஹவு எஃபெக்டிவ்லி எ ட்ரக் ஆர் ரிசெப்டார் பைண்ட்ஸ் டு தி how a drug or a chemical binds to the receptor, that is called as molecular docking. So it can be, you say, the small molecule can be there. And more important, ADMET. Why ADMET is important? Once you enter into the pharmacology, there will be two terms, pharmacokinetic and pharmacodynamic. What drug does to the body is called as pharmacodynamic. That dynamics the molecular docking. What body does to the drug in the sense kinetic, that ADMET property. Almost 39% of the drug which is withdrawn from the market is due to the toxicity or poor pharmacokinetic property. So always whenever you are doing, you should look into that and you have to make it out. So you should not stop with molecular docking or ADMET. There are one more term called as molecular dynamics. Because the docking, it will take place in an empty space where it won't have any hindrances, any disturbances, anything. But my body, our body is made up of water, right? Yes, it will be having motion, movement, always will be in movement. My body will have pressure, my body will have isotonicity, everything. How my body will work, based on that, if I try to explore, that is called as molecular dynamics simulation. Simulating like my body condition. So like this, each and every part of our body inside, it may look like a normal, but inside our body, this is the exact scenario which is happening. So I'll come into the topic called as AI. What is artificial intelligence? It's just like it's a field of computers, making computers to think like me. How I want, what I need, that's called as artificial intelligence. So example, artificial intelligence is a new term. No, Google is predictive. If I'm planning to go to Pondicherry with my friends or something else, if I type POND, only I have typed what it says. What is the temperature, what is the visits, where I should go, what are the things. This is called as predictive AI. Language translation. If I'm going to Karnataka, I don't know the language. I want to say wishes, good morning, good afternoon, what are something else. If I want to say in the Google will be translated and give. That is called as another AI. Face recognition, all knows. Only your mobile will predict your face and it will open. Sometimes without makeup, morning, if you try to open, will it open? கொஞ்சம் ஒரு பவுடர் போட்டு ஒரு ஃபவுண்டேஷனு ஒரு லைட்டாக ஒரு க்ரீமு பண்ணுவோம்ல, அது ஏன்? பிகாஸ் இட் ப்ரெடிக்ட்ஸ் ஒன்லி யூ. திஸ் இஸ் ஆல்சோ அ டைப் ஆஃப் ஏஐ. ஸோ virtual assistants, Amazon Alexa, iPhone Hey Siri, Hey Google, Hey Cortana. Hey Cortana went off long back. But these all are the another virtual assistants. If I say, only my mobile will, all the mobile, if you take Apple, the common term is Hey Siri. But only my mobile will open, yes boss. But your mobile will never open. That is called artificial intelligence. So email filtering, in your email you have seen, right? What are the things you have seen? Inbox will be there, updates will be there, promotions will be there, all the things will be segregated. This is also a type of AI. Image to text. So olden days your old senior notes will be there. You take a photo, upload to ChatGPT or some other tools, it will write and give, then you submit your assignment. That's also a OCR conversion. So I'll come to the story, history of AI. Without knowing the history, we should not speak. Whether the AI is a new term? No. It was coined in 1950s. போன ஜென்மம் நம்ம இருந்த மாதிரிலாம் தெரில, அந்த ஜென்மத்தில் அப்பயே வந்துச்சு டேர்ம் 1950 பக்கமே. ஸோ ஃபர்ஸ்ட் இன் 1942 பாம்பே மிஷின் வாஸ் ஐடென்டிஃபைட். பாம்பே மிஷின்றது போர்க்காலத்தில் World War II டைமில் யூஸ் பண்ணாங்க. எனக்கு எதிரி நாட்டுக்கு சண்டை நடக்குது, என் எதிரி நாடு வந்து அவனோட ஆட்களுக்கு ஏதோ தூது சொல்கிறான், ஆனால் என்னால் அது புரிஞ்சாதான் அவன் அடிக்க முடியும். அப்போ அது புரிஞ்சுக்கிறதுக்கு கொண்டு உருவாக்கப்பட்ட மிஷின் தான் பாம்பே மிஷின். அதை வச்சு தான் இது பண்ணாங்க. அவன் என்ன சொல்கிறான் இன்டர்பிரேட் பண்ணுறது. அங்கே ஸ்டார்ட் ஆனது 1942. In 1950 Alan Turing is the person. And then 1955 தான் ஜான் மெக்ரத்தி இது மாதிரி ஒரு கான்ஃபரன்ஸ் எங்கேயோ போகும்போது, ஐயா இது மாதிரி ஒரு பொருள் இருக்குது, இது பேர் ஆர்டிஃபிஷியல்னு காயின் பண்ணார். 1955 ஜான் மெக்ரத்தி இஸ் கால்ட் as the father of AI. Then 1961 இண்டஸ்ட்ரியல் ரோபோட் and first chatbot. கம்ப்யூட்டருக்கு ப்ரோக்ராம் பண்ணி கொடுத்துறாங்க கேம் ஆடுறதுக்கு. செஸ் வேர்ல்டு சாம்பியனுக்கும் அந்த கம்ப்யூட்டருக்கும் சண்டை. பட் கம்ப்யூட்டர் won the match. That was happened in the year of 97 and 98. Kismet emotionally equipped robot. எல்லாரும் இந்த படம் பார்த்துறீங்களே, சிட்டி ரோபோ, எந்திரன் 2, சனா 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 எஸ். ஸோ எமோஷ்னலி எக்யூப்டு, எமோஷன்ஸ் புரிஞ்சிக்கக்கூடியது. அது எப்போ 98லேயே கொண்டு வந்துட்டாங்க. நம்மால் சங்கர் அது அப்போ எப்போ பார்த்தாருன்னு தெரியல, வழக்கம் போல காப்பிச்சு விட்டார். ஸோ 2002 கிளீனிங் ரோபோட். இந்த என்ன மாதிரியா உங்களை மாதிரியா வீட்டில் பொண்டாட்டி ஒருத்தவரை தாங்க முடியாமல் ஒரு மிஷினை கண்டுபிடிறா தொடச்சி வர்றதுன்னு கண்டுபிடிச்சது தான் கிளீனிங் மிஷின். இது இட் வில் பி அப்ளிகபிள் ஃபார் போத் மேன் அண்ட் ஃபீமேல். யார் வேணாலும் கண்டுபிடிச்சிடலாம். நீங்கள் கஸ்டமருக்கு நீங்கள் கண்டுபிடிச்சிடலாம், இல்லை நீங்கள் அடிச்சடியை பார்க்க தாங்க முடியாமல் ஆம்பளையும் கண்டுபிடிச்சிடலாம். ஸோ ஐ டோன்ட் நோ ஹூ ஹவ் ஐடென்டிஃபைட். ஸோ in the year of 2008 I3 that recognition came. So then Jeopardy, Ampere, 2018 GPT, ChatGPT is there and GPT, generative pre-trained models, generative pre-trained transformers. They are the things already it will be trained and it will be utilized. This is how a process goes on. And this ChatGPT came in 2018, but after COVID we are using more. So now you know n number of tools. After 2022-23 onwards, there are cages and cages of tools are being launched daily. So this is the AI. So this AI, how we are going to utilize in our pharma field, because it's an application. We need some application to improvise, fasten the process. So these are the basic things, applications which is used in the pharma sector. One is for drug development, drug discovery, இந்த மாதிரி ஜென்ரல் ப்ராசஸ் நம்ம ஃபஸ்ட்டு பார்த்தோம்னா அந்த எல்லாத்துமே தேவைப்படும். தென் கிளினிக்கல் ட்ரையல்ஸில் பிஸ்னஸ் இன்டெலிஜென்ஸ் எந்த இடத்துல என்ன மார்க்கெட் போகுதுன்னு பேஸ் பண்ணி ஒர்க் அவுட் பண்ணுவாங்க. ஸோ வாட் இஸ் த அட்வான்டேஜஸ் ஆஃப் ஏஐ? More powerful and more useful than of the computers, new and improved interfaces, solving new problems. That's the most important. Better handling information, release information overload and this thing. ஸோ நீங்கள் எல்லாருமே ஹாஸ்பிட்டல் யாராவது அட்மிட் ஆகி வெளில டிஸ்சார்ஜ் ஆகும்போது they will be maintaining a record. இந்த மாதிரி இன்னார் வந்தாங்க, இந்த கண்டிஷனோடு வந்தாங்க, இவங்களை இந்த ட்ரீட்மெண்ட் பண்ணோம், இப்படி ஆச்சு, இன்னைக்கு டிஸ்சார்ஜ் பண்ணுறது. அது மெடிக்கல் டாக்குமெண்டேஷன் ஃபைல். அந்த ஃபைல் என்ன அந்த காலத்தில் கையில் எழுதி அது அப்படியே ஒரு மெடிக்கல் டாக்குமெண்டேஷன் ரெக்கார்ட் ரூம் இருக்கும், அங்கே போய் வச்சுருவாங்க. அதனால் யாருக்கும் அந்த பயன் இல்லை. அதை டேட்டாவை எடுத்து கன்வெர்ட் பண்ண நாலேஜாக இப்போ உள்ள கண்டிஷனுக்கு எனக்கு அதை பற்றி ஐடியா இல்லை. ஸோ இந்த மாதிரி ஒரு பேஷண்ட் வந்திருக்கான், நான் என்ன மாதிரி ட்ரீட்மெண்ட் கொடுக்கலாம், என்ன மாதிரியான கண்டிஷன் பார்க்கலாம், அந்த மாதிரி ப்ராசஸ் தான் ஏஐ வந்து நவ் இட்ஸ் மோர் யூஸ்டு. Conversion of information to knowledge. So SCR, QSR and machine learning. This is the three things. SCR first step, அந்த இடத்துல என்ன ஸ்ட்ரக்சர் இருக்கணும், டோனாக இருக்கணுமா, அக்செப்டாக இருக்கணுமா, எப்படி ஆக்டிவிட்டி தரும். QSR அதில் மினிமலாக எந்தெந்த ப்ராப்பர்ட்டி எத்தனை குவான்டிட்டி இருக்கணும்னு. நெக்ஸ்ட் லெவல் தான் மிஷின் லேர்னிங். அது எத்தனை ப்ராப்பர்ட்டி வேணாலும் இருக்கலாம், எப்படி வேணாலும் இருக்கலாம், அதுலேருந்து நான் பெஸ்ட் மாடல் கொடுப்பேன், அப்படிதான் மேட்ரு. First generation is SCR, second generation is QSR, third generation is machine learning or AI models. So machine learning on the computers are, we are making the computers to automatically think and process. That's the only major thing which we will make the system to behave like a human. நம்ம கிட்ட ஒரு நாய்க்குட்டி வந்து நம்மள மாதிரி மாற்றோம் பார்த்தீங்களா, அதே தான் கம்ப்யூட்டர்ஸ். இதில் வந்து 3 முக்கியமான விஷயம் இருக்குது. only 3 things we have to look into it. One is task, experience and performance. Task means நான் என்ன வேலை பார்க்க போகிறேன், எனக்கு என்ன தேவை, அந்த தேவைக்கு வந்து இதுவும் பழைய டேட்டா இருக்கா, அது எக்ஸ்பீரியன்ஸ். அப்புறம் பர்ஃபார்மன்ஸ், இதை பேஸ் பண்ணி எனக்கு எப்படி பர்ஃபார்மன்ஸ் டேட்டா வருது. டாஸ்க்கு எனக்கு என்ன தேவை, இதுக்கு எதுக்காக டேட்டா இருக்கா, நான் என்ன மெத்தட் யூஸ் பண்ண போகிறேன். இவ்வளோ தான் மிஷின் லேர்னிங்கோட மாடல்ஸ். ஸோ இந்த டெக்னிக்ஸ் அதில் இப்போ ஏற்கனவே சொன்ன மாதிரி லைக் மாலிகுலர் டாக்கிங், ஃபார்மகோஃபோர் மாடலிங், சேம் வே மிஷின் லேர்னிங் வில் ஹேவ் 4 major objectives. One is supervised, unsupervised, semi-supervised and reinforcement. Supervised in the sense, if I'm going for a working, I should have a complete data. I'll show us an image. Yes, supervised learning in the sense, I'll be having a label and I'll be having the data. எனக்கு வந்து ஒரு இதுதான் ஆப்பிள், இதுதான் ஆரஞ்சு, இதுதான் கேரட் அப்படின்னு எனக்கு சொல்ல தெரியும். எனக்கு இந்த பொருளாக இருக்குது, இதை செக்ரேட் பண்ணி கொண்டு சூப்பர்வைஸ். ஐ எம் சூப்பர்வைசிங் யூ. உனக்கு ஒரு கைடு இருந்து ஒர்க் பண்ண எப்படியோ அந்த மாதிரி தான் சூப்பர்வைஸ். அப்போ என்ன ஆகுனா, ஸோ லேபிள் டேட்டா, லேபிள்ஸ். ஸோ ஆட்டோமேட்டிக்லி இட் வில் ட்ரெயின் அண்ட் ப்ரெடிக்ஷன். இஃப் ஐ கிவ் அ எக்ஸாம்பிள், what it will show, it will carrot, it is bell pepper, it is tomato. This is called as supervised learning. Unsupervised, it doesn't have any idea because there is no previous past record. There is no record how it will work out. So based on the shape and structure, if you see here, green pepper and red pepper are, bell pepper are in one size. Unsupervised in the sense, automatically what it thinks, based on that it will do off. So reinforcement in the sense, how you perform based on that results. Now there is a water bottle or something else. If I walk towards that, then only I'll get the benefit. So that the change in the values, adjustment, then only the fruitful result will come. This is reinforcement. Then, and the last one is unsupervised model. Sorry, semi-supervised model. So in the semi-supervised model, a little data I know, not completely, but if I give, it will try to predict the best out of the best. So here I have given the label of orange and banana, but unlabeled, it will predict as a apple. So this is how it will work. Supervised, semi-supervised, unsupervised and reinforcement. So in this n number of models will be there. Neural network, how our brains are there, they are the methods. And then decision tree in the sense, I'll be calling my friend, shall we go for a outing? மழை வருமா வராதா, வந்தால் என்ன பண்ணலாம், வரலன்னா என்ன பண்ணலாம், சரி மழை வரல வெளில போகலாமா, எங்கே போகலாம், அந்த மாதிரி decision, if you keep on taking then it is called as a decision tree. Then random forest வந்து ஒரு பர்டிகுலர் ஃப்ரெண்டோட பேசி எடுத்தேன்னா அது டிசிஷன் ட்ரீ. நான் 4-5 ஃப்ரெண்டோட பேசி உங்கூட என்ன ப்ரோக்ராம் எடுத்தேன்னா that is called as a random tree models. And then support vector machine, it will be dividing the content into 3 phases. And then the next one is KNN, K nearest model. It's simple like rounding of the figure 1.56. அப்போ என்ன பண்ணுவோம், 1.6ன்னு எழுதுவோம், 1.54, 1.5 எழுதுவோம். அந்த மாதிரி இது ஃபஸ்ட்டு டேட்டா செக்ரிகேஷன் அகுமெண்டேஷன் பண்ணிவிட்டு பண்ணும். ஸோ ஒரு ஆட்-அப் பூஸ்ட், சின்னதாக லைட்டாக ஒரு புஷ், அந்த ஓட்டபிக்கால் எல்லா டேட்டாவும் யூட்டிலைஸ் பண்ணியிருக்கோம். ஒரு 10 நல்லா இருக்குது, 8 நல்லா இல்லை, மறுபடியும் 9 நல்லா இல்லை அப்படின்னு வரும்போது ஈக்குவலாக 9 கொடுத்துட்டு ஈக்குவலைஸ் பண்ணி அனுப்பிச்சுட்டோம். ஸோ do you have any questions? As I said, 30 minutes done. Any questions? எப்படா சோறு கொடுவீங்க? So I would like to thank all the participants, MS Raman University of Applied Sciences, JKK Sambhurna College of Pharmacy, and special thanks to Venkatachalam sir, who is my friend and a long-term collaborator, and this is my lab. If you need any support in the help of computational things, you can have anytime you can approach us. Thank you for your attention.
Hello. Thank you, sir. Thank you, Parasuraman sir. Please, sir. Sir is entirely covered from 1962 to 2021. in the field of drug discovery process. He started in silico method from 1962. The first CAD software was started. Now this generative AI 2021, up to that, all the developments areas he's touched, is explained very nicely. Thank you so much, sir, for this. Your information is very good. Our students definitely will have some points. Then many partly you are given some softwares also. What are the areas, what are the free softwares are there, all the things you are given to us. Thank you so much, sir, for your valuable session. Thank you, sir.
Thank you, sir. Now I request Chairperson Dr. P. Balan, sir, to honor the eminent speaker, Dr. Parasuraman, sir. Thank you, sir. Now I request Dr. K. Sumadi ma'am to honor the chairperson, Dr. P. Balan, sir. Thank you, ma'am. Dear delegates and students, delicious lunch will be provided near hostel. Our volunteer will guide you for I would like to announce e-poster presentation winners. The first prize is in e-poster presentation is shared by 3 participants. First prize goes to Mugilarasi C, Vinayaka Mission College of Pharmacy, Salem. Puspa Venkatesh, Vivekananda Pharmacy College for Women. Third person, Madhumati D, Annai College of Pharmacy. The second prize in e-poster presentation is shared by 3 participants. R.D. Jaykumar, Padmavati College of Pharmacy, Dharmaburi. K.S. Praveen, JKK MMRF and JKK Samburnyamal College of Pharmacy. K.S. Praveen. Monica Charles A, Sri Rangapubadi College of Pharmacy. Monica Charles. Third prize in e-poster presentation is shared by 3 participants. Tarun A.M., Pavai College of Pharmacy. V. Ganapati, KG College of Pharmacy. Abubakar Siddiq, Erode College of Pharmacy. Now I would like to announce oral presentation winners. The first prize is in oral presentation is shared by 3 participants. M. Tarun, JKK MMRF and JKK Samburnyamal College of Pharmacy. Idanadu Jesslyn Sneha, SVCP Tiruchengodu. Keerthana, Nanda College of Pharmacy. And the second prize in oral presentation is shared by 3 participants. Second prize, Indumadi, Nanda College of Pharmacy. Jyotika, SS Institute of Pharmacy. College of Pharmacy. Third prize in oral presentation is shared by 3 persons. Sandhya, Sri Shanmuga College of Pharmacy. Induja, JKKM, TN Palayam. Sandhya, Sri Shanmuga College of Pharmacy. Induja, JKKM, TN Palayam. Kalaiselvi, Sri Ram Nallamai Yadav College of Pharmacy. Sri Ram Nallamai Yadav College of Pharmacy. I kindly request student and one faculty from KSR College of Pharmacy to receive their certificates. I kindly request student and one faculty from KSR College of Pharmacy to receive their certificates. Students and faculty from Abhirami College of Pharmacy. Abhirami College of Pharmacy. Students and faculty from Annai College of Pharmacy. Annai College of Pharmacy. Annai College of Pharmacy. I request students and faculty from Cheran College of Pharmacy. Cheran College of Pharmacy. Erode College of Pharmacy. Erode College of Pharmacy. Erode College of Pharmacy. I request students and faculty from Excel College of Pharmacy. Excel College of Pharmacy. Excel College of Pharmacy. I request students and faculty from Powai College of Pharmacy. Powai College of Pharmacy. Powai College of Pharmacy. I request students and faculty from Elithachan College of Pharmacy, Trivandrum. Elithachan College of Pharmacy. I request students and faculty from KM College of Pharmacy, Madurai. of Pharmacy. I request student and faculty from KG College of Pharmacy, Viluppuram. KG College of Pharmacy, Viluppuram. I request students and faculty from Mahendra College of Pharmacy, Namakkal. KG College of Pharmacy. I request students and faculty from Mahendra College of Pharmacy, Namakkal. Mahendra College of Pharmacy. I request students and faculty from Nanda College of Pharmacy, Erode. Nanda College of Pharmacy, Erode. Mahendra College of Pharmacy. Pharmacy. I request students and faculty from Padmavati College of Pharmacy, Dharmaburi. College of Pharmacy. faculty from SNS College of Pharmacy, Coimbatore. SNS College of Pharmacy. I request students and faculty from Rangabhupati College of Pharmacy, Viluppuram. Rangabhupati College of Pharmacy, Viluppuram. Rangabhupati College of Pharmacy. RKB College of Pharmacy. I request students and faculty from RKB College of Pharmacy, Krishnagiri. Rangabhupati College of Pharmacy. RKP College of Pharmacy. I request students and faculty from Vinayaka Mission College of Pharmacy, Salem. Vinayaka Mission College of Pharmacy, Salem. I request students and faculty from SS Institute of Pharmacy, Sangagiri. SS Institute of Pharmacy. SS Institute of Pharmacy. I request students and faculty from SNS College of Pharmacy. I request students and faculty from Sri Ram Nallamani Yadava College of Pharmacy, Tenkasi. Sri Ram Nallamani Yadava College of Pharmacy, Tenkasi. Sri Ram Nallamani Yadava College of Pharmacy, Tenkasi. I request students and faculty from Sasuri College of Pharmacy, Perundurai. College of Pharmacy. I request students and faculty from Sri Shanmuga College of Pharmacy, Tiruchengodu. Sri Shanmuga College of Pharmacy. Students and faculty from SSM College of Pharmacy, Jambai. SSM College of Pharmacy, Jambai. SSM College of Pharmacy, Jambai. I request students and faculty from Swami Vivekananda College of Pharmacy, Tiruchengodu. Swami Vivekananda College of Pharmacy, Tiruchengodu. I request students and staffs from Vijay Vidyalaya College of Pharmacy, Dharmaburi. Vijay Vidyalaya College of Pharmacy, Dharmaburi. Faculty from Vivekananda College of Pharmacy, Sangagiri. Vivekananda College of Pharmacy, Sangagiri. Vijay Vidyalaya College of Pharmacy, Dharmaburi. of Pharmacy, Sangeri.
vote of thanks சொல்ல சொல்லாங்களா மேடம்?
Let's conclude the seminar with the national anthem. I request everyone to stand.